करेगा बोलेंगे अच्छे से
तो स्ट्रीमिंग स्टार्ट हो गया सर
काम कीजिए इसको रखे रहेंगे
लगा काम पड़ेगा तो फिर हम
उसी से ले लीजिएगा अनाउंसमेंट लिखा हुआ है
एक अनाउंसमेंट प्रोग्राम दीजिए उसको
Good morning, students. I'm Dr. Emma, your HOD. I'm going to announce certain instructions. Please follow them. All the participants, please mute your audio and disable your video. You are requested to use earphone or headphone during the session. Do not try to share your device's screen. It will hinder the event. Type your questions in the chat box
after the end of the
session.
Fill up
your feedback form, which will be provided to you in the third session.
Right
now, I'm requesting you to mute your audio and disable your video.
One
girl, your
video camera
is on.
Kindly
disable it.
Thank you.
Thank you.
Dina
Sunaina, kindly unmute yourself and disable your video.
After announcing the pregnancy, पार्टी चालू है अरे कैसी भोजपुरी गाने हैं ना उनको दिखाएंगे फेसबुक में आया इसे पूरा डब कर दिया अनुष्का विराट का नया समाचार वाला
Kindly unmute yourself. Somebody's mic is on. Dina, Sunaina, please
disable your
video. Sonamai.
Anshu, Archana, disable your video.
I muted everyone.
तो अनम्यूटेड है लेकिन ना फिर भी
नहीं म्यूट कर दिया म्यूट कर दीजिए
ये सोनामाई देवगम शी इज स्टार्टिंग वीडियो ना ये दोबारा की
ये इंटरनेट के चलते फिर से यूट्यूब पर में नहीं चल रहा है वीडियो
अच्छा मीटिंग तो कंटिन्यूअस वाला है ना
हाँ मीटिंग कंटिन्यूअस है
क्योंकि रिकॉर्डिंग लिखा हुआ था
हाँ नहीं मीटिंग कंटिन्यूअस है
देखिए ना निकी एंजल ज्वाइन की है
नहीं
की है नहीं ना बाबा वेरी शी हेलो Nikki, you have not joined. Where are you? You have not joined? Hurry up. Hurry up.
इंटरनेट के चलते ये नहीं काम कर रहा है यूट्यूब वाला. The internet connection is low, that's why we are not getting the suitable video. नेट का ये है अब इशू.
एक बच्ची उस दिन तो याद है ना आपको, एक बच्ची इंट्रोडक्शन पढ़ेगी रिसोर्स पर्सन का, फिर रिसोर्स पर्सन बोलेगा, बाकी सब सेम चलेगा. क्या हुआ? फिर बंद हो जा रहा है मेरा बीच बीच में, पता नहीं क्यों.
मौसम के चलते ये हो गया है.
भगवान मौसम चुप रहे, हो चाहिए.
यूट्यूब का तो चल ही नहीं रहा है. यूट्यूब वाला चल ही नहीं रहा है.
यूट्यूब स्ट्रीमिंग नहीं हो रहा है.
नहीं मेरा मतलब इसके चलते नहीं हो रहा है.
राउटर से चला सकते हैं.
तो पासवर्ड
राउटर का पासवर्ड डब्बा होगा, कोई चीज तो दिखाया नहीं. अभी डेटा फिलअप होगा क्या? डेटा फिलअप करने से होगा नहीं?
डेटा है ही मेरा.
है.
हाँ, हम कल ही रिचार्ज करवा लिए 51 वाला कि अलग से अगर मान लीजिए कि अगर काम पड़ेगा तो उसके लिए, लेकिन नेटवर्क ही नहीं काम कर रहा है आजकल. वीडियो का रिजोल्यूशन अगर कम कर दें तो होगा.
क्या करूं?
मान लीजिए वीडियो का रिजोल्यूशन हम थोड़ा सा कम कर देते हैं,
हाँ.
हो सकता है तो वो करेंगे तब तो फिर से लिंक क्रिएट करना होगा यूट्यूब का.
यहाँ 100 बच्चे तो एंटर कर सकता है.
हाँ यहाँ कर जाएगा.
तो ठीक है, 100 बच्चे देख लेते हैं, अधिक होता है तो 56 ही तो हुआ है, एंटर करने दीजिए. ट्राई, जब भी हो सकता है उसी वक्त से शुरू कर दीजिए।
नहीं, स्ट्रीम कर रहा है वो वहां पर, वो स्ट्रीम कर रहा है, मेरा इसमें दिखा रहा है कि स्ट्रीम कर रहा है, लेकिन लिख रहा है कि ड्यू टू, यहाँ पर मैं ये दिखा रहा है that due to stream health the resolution is not working over here। नहीं, ड्यू टू स्ट्रीम हेल्थ, मतलब स्ट्रीम नहीं कर पा रहा है ठीक से। अभी लेकिन हम आपके वाले रूटर से भी कनेक्ट नहीं कर पाएंगे, क्योंकि हम जैसे ही कनेक्ट करेंगे मेरा इससे पहले हटेगा, और उसे हटेगा तो ये पूरा खत्म हो जाएगा मीटिंग। अभी मतलब अगर रूटर का पासवर्ड दीजिएगा ना, तो हम जैसे ही इसको कनेक्ट करने के लिए जाएंगे तो पहले ये इससे हट जाएगा और वो पूरा जूम मीटिंग हट जाएगा।
अभी क्या चल रहा है कि नहीं चल रहा है?
नहीं, स्ट्रीम उधर हो रहा है।
हाँ।
ये देखिए, ये दिखा रहा है कि लाइव हो गए हम 12 मिनट 50 सेकंड से, लेकिन यहाँ पर मैं दिखा यही नहीं कर रहा है। वीडियो रिजोल्यूशन बोल रहा विच इज नॉट ऑप्टिमल। ये जो स्ट्रीम स्टेटस आता है ना, ये एक्सीलेंट हो जाता है, ग्रीन कलर हो जाता है, तब वो स्ट्रीम करना स्टार्ट कर देता है। लेकिन सबको मैसेज कर रहे हैं वहां पर ना, लाइव चैट। स्ट्रीम तो हो ही गया है। ठीक है, मेरे को पता नहीं क्यों नहीं दिखा रहा। ओहो,
पुराना वाला कर देते
हम
हैं।
पुराना वाला खोल देते हैं।
इतना दिक्कत हुआ है हमको जल्दी
हाँ।
जल्दी।
ठीक है, हम कर रहे हैं।
आगे से सही है। किसी को चाहिए था? मैं ढूंढने की कोशिश कर रही हूँ। हाँ नमस्कार सर, प्लीज ज्वाइन कर लीजिए मीटिंग। हाँ प्लीज। sister. Yes, sister, please bless us for the fruitful workshop. Yes, first you please bless us. Yeah. Thank you, sister. Thank you, sister. Our first resource person will be reaching within 3 minutes, sister, because of certain road jam. So we will start at time only, sister. Thank you very much, sister. Thanks, stay with us. Thank you. Thank you, sister.
कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः शत्रुबुद्धिविनाशाय दीपज्योतिर्नमोऽस्तुते दीपज्योतिर्नमोऽस्तुते शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः
बैकड्रॉप चल रहा है मेरे को नहीं आ रहा है
कुछ हो रहा है। किनका बोले थे कौन ऐड नहीं हुआ?
हाँ
कौन ऐड नहीं हुआ?
जेबी पानी ऐसे भी चीजें
नहीं ऐड हुआ।
क्योंकि मैं तो सेशन स्टार्ट ही नहीं कर सकती थोड़ा सा मेरा स्क्रीन देख लीजिए तो
नहीं आया आपका? लाइटनिंग ऑफ लैम्प जब करना होगा तो मैम थोड़ा ये कर दीजिएगा। नहीं मतलब इशारा कर दीजिएगा।
हाँ
दस तीन हो गया, 10:03 हो गया।
बाकी सब पूरे रुक जाएगा। Meeting would be started at 10. Okay, if somebody 9:53.
ये किसी दूसरे नाम से तो नहीं ना ऐड हो गए हैं?
नहीं वो ही नहीं है, वो रोड में ट्रैफिक था तो जाना पड़ता है।
क्योंकि इसमें रेडमी, रियलमी ये सब भी है। इस नंबर कोई 769939 करके भी है।
To wait patiently for some 3-4 minutes. Here, our resource person will be joining, and then we will start the program. Sister Principal has joined. Welcome her. Okay? Pardon? Couldn't understand. Yeah, everybody is muted, but your sound will go now.
वो कर दो।
Yeah, it will be done for that moment, and then again they will be muted. ठीक है? Wait for certain seconds. Okay, Anand? Now you see.
session will start shortly. Please wait patiently for more 5 minutes. Our esteemed resource person is on traffic jam, so we'll be waiting for him. Thank you.
Hi, sir. Okay, you have joined. Have you joined? Okay, okay. Okay, thank you, sir. We are starting. Okay. Yes. सर मेरे को अनम्यूट कर देंगे तो
हाँ कर लीजिए, आप अनम्यूट कर लीजिए.
Hello, Nikki. Hello.
Yes.
Nikki,
Yes.
am I audible to you?
Please start
Please
the
Yes,
start the
ma'am.
program.
Please start the
Please start
program.
the program.
Okay, ma'am. Okay, ma'am. Respected Principal, Nirmala College, Ranchi, Dr. Sister Jyoti, Vice Principal, Sister Shobha, our Honorable Resource Person, Dr. Jay Prakash Pandey, CTR-TI Senior Scientist, Dr. Emma R.
वो
Seraphim,
जब बोलेंगे
HOD
तब
of
रुकता है.
Department of Zoology, Nirmala College, Ranchi, Dr. Anubhuti Singh, Department of Botany, and my dear friends, a warm good morning to all. On behalf of Department, Botany and Zoology, with immense joy and pleasure, I welcome our dignitaries and participants. Today, we have gathered here to organize our one-day workshop on exploring the science behind sericulture, endocrinology, and ethnobotany. In the beginning, God said, "Let there be light," and there was light. God saw that the light was good, and he separated the light from the darkness. Genesis chapter 1, verse 3. So let us remember God by the lighting of lamp. May I request our principal, Dr. Sister Jyoti, Nirmala College, Ranchi, Vice Principal, Sister Shobha, Nirmala College, Ranchi, Honorable Resource Person, Dr. Jay Prakash Pandey, CTRTI, Senior Scientist, Dr. Emma R. Seraphim, HOD of Department of Zoology, Dr. Anubhuti Singh, Department of Botany, for the lighting of lamp.
शुभं कुरु त्वं कल्याणम् आरोग्यं धनसम्पदः शुभं कुरु त्वं कल्याणम् आरोग्यं धनसम्पदः शत्रुबुद्धिविनाशाय दीपज्योतिर्नमोऽस्तु ते दीपज्योतिर्नमोऽस्तु ते शुभं कुरु त्वं कल्याणम् आरोग्यं धनसम्पदः शुभं कुरु त्वं कल्याणम् आरोग्यं धनसम्पदः
May I request our HOD of Zoology Department, Nirmala College, Ranchi, Dr. Emma R. Seraphim, to bless us immensely so that we may be enlightened with the stream of knowledge. Over to ma'am.
Thank you, Nikki. Respected Principal, Nirmala College, Ranchi, Dr. Sister Jyoti, respected Vice Principal, Sister Shobha, esteemed resource person, Dr. Jay Prakash Pandey, Senior Scientist, CTR NTI, dear colleagues and my dear students, a very warm good morning to you all. With great joy and love, I welcome you all to this online students workshop on exploring the science behind
Sericulture,
endocrinology and ethnobotany,
organized by
Botany
Botany
and
and
Zoology
Zoology
Department
Department
of
of
Nirmala
Nirmala
College,
College,
Ranchi.
Ranchi.
Students,
Students,
a
a
workshop is a
workshop is a
platform
platform
where
where
people
people
barter
barter
knowledge
knowledge
and
and
experience.
experience.
It is
It is
truly
truly
said,
said,
the
the
more you
more you
learn,
learn,
the more
the more
things you will
things you
know.
will
The more you read,
read,
the more
the more
places you will
places.
go. Today we
Today
have
we have
gathered
gathered
to
to
talk
talk
over
over
the
the
three
three
different
different
but
but
curriculum-based
curriculum-based
topics
topics
with
with
eminent
eminent
resource
resource
persons.
persons.
This
This
workshop is,
workshop
in
is, in
fact,
fact,
the
the
brainchild of
brainchild
our
of our
dynamic
dynamic
principal,
principal,
Dr. Sister
Dr. Sister
Jyoti.
Jyoti.
It is
It
her
is her
support
support
and
and
motivation
motivation
that has
that
made
has
the
made the
organization of
organization
this
of this
event
event
a
a
successful one.
successful one.
I must
I must
let you know
let you know
that
that
Botany and
Botany and
Zoology
Zoology
Department of
Department of
Nirmala
Nirmala
College
College
are
are
running
running
DBT
DBT
Start
Start
College
College
scheme
scheme
under
under
DBT
DBT
project.
project.
Both the
Both the
departments
departments
owe their
owe their
every
every
success
success
to their
to their
beloved
beloved
principal,
principal,
Dr. Sister
Dr. Sister
Jyoti.
Jyoti.
Dear
Dear
students,
students,
it is my
it is my
heartfelt
heartfelt
desire
desire
that
that
you all
you all
take
take
this golden
this golden
opportunity
opportunity
to
to
learn
learn
and grow,
and grow,
attain
attain
such
such
prodigious
prodigious
heights
heights
that you
that
will
you
make
will make
Sister
Sister
Principal
Principal
as well
as
as the
well as the
institution
institution
proud.
proud.
You all
You all
have
have
enhanced our
enhanced our
hopes
hopes
as
as
you have
you have
successfully
successfully
taken part
taken part
in the
in the
brochure
brochure
making
making
competition.
competition.
You are
You are
conducting
conducting
this
this
workshop.
workshop.
This
This
also
also
proves your
proves your
mantle.
mantle.
At
At
last,
last,
I
I
request you
request you
to
to
participate in
participate
the
in the
workshop
workshop
with whole
with whole
heart,
heart,
mind,
mind,
and
and
might.
might.
May the wind
May the wind
rise
rise
beneath your
beneath your
wings
wings
and carry
and
you
carry
to the
you to the
heights of
heights of
success.
success.
May our
May our
blessings
blessings
guide you
guide
to
you
your
to your
goals.
goals.
With
With
lots of
lots of
best
best
wishes,
wishes,
I
I
ask you
ask you
to
to
participate
participate
in this
in this
workshop.
workshop.
Thank you.
Thank you.
Thank you, ma'am. With immense pride and exultation, may I request our motivating and guiding light, Dr. Sister Jyoti, Nirmala College, Ranchi, to address this online workshop. May we be blessed for the fruitful workshop. Over to Principal.
Thank you, Nikki. A very warm and blessed good morning to one and all. Respected Vice Principal, Sister Shobha, our esteemed resource person of the day, Dr. J.P. Pandey, Senior Scientist, CTRT Ranchi, respected HODs, Dr. Emma Rani Seraphim, and Dr. Anubhuti Singh, entire faculty members of both the departments, botany and zoology, all other faculty members of Nirmala College and others, and my dear students. First of all, I thank the Almighty for giving us this graceful day with numerous blessings. Also thank Him for giving us knowledge and wisdom to sustain and nurture the same with various means. May the Almighty shower His blessings upon each one of us and upon the entire humankind.
म्यूट कर रहे हैं या अनम्यूट कर ले रहे हैं अपने आप को? बार बार म्यूट कर रहे हैं ये लोग, अपने आप को अनम्यूट कर ले रहे हैं। देखिए, हम म्यूट कर रहे हैं, ये लोग अनम्यूट कर ले रहे हैं। सबका म्यूट हो जा रहा है, सबका म्यूट हो जा रहा है। इनको हम निकाल दे रहे हैं बाहर से। ऐसा हम आप क्या कर सकते हैं? इनको निकाल दे रहे हैं। मैम को मैं बोलिए ना अनम्यूट करने के लिए। मैम म्यूटेड।
निकी सिस्टर को अनम्यूट करने के लिए पूछ लीजिए।
Excuse me, sister. Sister, kindly unmute yourself.
All the girls unmute yourself. Mute yourself. Do not unmute. Mute yourself.
I could hear many voices.
Thank you, sister.
Okay, so once again I begin, okay?
Yes, sister, please.
A very warm and blessed good morning to one and all. Respected Vice Principal, Sister Shobha, our esteemed guest and the resource person of the day, Dr. J.P. Pandey, Senior Scientist, CT/RT, Ranchi, respected HODs, Dr. Emma Rani Seraphim, Dr. Anubhuti Singh, and the entire faculty members of both the department, Botany and Zoology, all faculty members of Nirmala College and others, and my dear students. First of all, I thank the Almighty for giving us this graceful day with numerous blessings. We also thank Him for giving us knowledge and wisdom to sustain and nurture the same with various means. Am I audible? Emma, am I audible? Am I audible? Nikki, yes?
Yes, sister. Yes, sister, you're audible. Yes, sister.
Good morning, the Almighty shower His blessings upon each one of us and the entire human community. Due to COVID-19 pandemic and subsequent lockdowns, we have been forced to remain indoors for about more than 5 months. But I want to assure you that we are connected through spirit. And I want to also assure you that we care for you, because we are one and belong to one bigger family of Nirmalites. And together, we move towards new horizon, keeping our motto aligned, one heart, one mind. Someone has rightly said, challenges are what make life interesting. Overcoming them is what makes life meaningful. Yes, our lives seem to be beautiful because we meet with challenges that in many ways shapes, reshapes, and molds us. At least it gives us the strength to face the adversities. The present condition has made everyone awestruck with despair. I can very well visualize that adapting to the ways of online teaching learning is the greatest challenge for a student in today's condition. But we at Nirmala College want to groom our students, you dear students, for every situation, whether easy or difficult. Nirmala College, Ranchi, affiliated to Ranchi University, Ranchi, a college with potential for excellence, accredited by NAC with grade A, constantly strives towards empowering students by imparting skill-based, knowledge-based programs. And today's workshop for students is one among such initiatives on the topic exploring the science behind sericulture, endocrinology, and ethnobotany. I am delighted to state that this lockdown period is the period of innovation and creative learning, as even during this period, online activities have been carried out to keep the students active and motivated. we offer our students the best possible opportunities to become confident, thoughtful, young people who are prepared for any future challenges in an exciting and increasingly global world. I deeply appreciate the commendable efforts and commitment of Dr. Emarani, Dr. Anubhuti, and all the faculty members of Department of Botany and Zoology making efforts to arrange such online workshop that will fulfill the academic demands of students in a profound manner. Push yourself because no one else is going to do it for you. So my dear students, make the best use of the opportunity of learning that is being provided to you. Education is the only path that has the capacity of changing lives of not just an individual, but of a generation. I hope this workshop proves to be an excellent learning experience for you all. Moreover, before I conclude, I congratulate the winners of the brochure making competition and acknowledge the efforts of our students in designing of the brochure. Certainly, it was a very innovative and creative idea indeed. Much appreciated. Congratulations to all faculty members of Botany and Zoology. My prayers and best wishes for a successful workshop that would definitely add to academic flavor of our students. May you have a pleasant time. May God bless you all. Thank you.
Thank you, sister. Thank you very much. Over to Nikki.
Thank you so much, Sister Principal, for your blessings and prayers. So, we will have 3 sessions today. First session will be on the topic Sericulture, and we have with us Dr. Jaiprakash Pandey, CTRTI Senior Scientist. Now I request Neha Kumari, Semester 3, Department of Zoology, to introduce our esteemed resource person, Dr. Jaiprakash Pandey, CTRTI Senior Scientist.
Am I audible to all?
Yes.
A very good morning to all. Honorable Principal, Nirmala College Ranchi, Dr. Sister Jyoti, Vice Principal, Sister Shobha, our esteemed resource person, Dr. J.P. Pandey, Head of Zoology Department, Nirmala College Ranchi, Dr. Emma R. Serakan, Dr. Anubhuti Singh, Department of Botany, all dignitaries and my dear friends. I welcome you all with a great joy and immense exultation today. We are pleased to welcome Dr. Jaiprakash Pandey sir, a very special guest from Central Tasara Research and Training Institute, CTRTI, Pisakanagari, Ranchi. Sir is a renowned person in the field of sericulture, designated as a scientist D, and shown his excellence or expertise knowledge in the field of molecular physiology of Tasar silkworm. Sir has done 55 publications and 11 citations. Sir, now I don't think so that you hardly need any more introduction. Your work talks a lot about your effort in the field of sericulture. Sir, feel privileged to continue the session ahead. Sir, please.
Excuse me, sir. Kindly start the session.
Yes. Hello.
Yes, sir. Please start the session.
Very good morning to you all. I am very thankful for Nirmala College. Yeah. Am I audible, madam?
Yes, sir.
Hello. Hello. Madam?
Yes, sir, you are audible.
Yes, sir.
Yeah. Thank you very much. I am very thankful to Nirmala College and especially our principal.
नहीं अनम्यूट अगर नहीं कर रहा है ना तो सब कोई अपना माइक्रोफोन ऑन कर ले रहा है
Today I am going to tell about what is the science behind sericulture. That is a very relevant topic. As associated with this Tasar sericulture industry, and as a student, and it is well established that after agriculture, the Tasar sericulture and sericulture is providing maximum job in our country. And the more importantly, this job is very important for society.
सर कुछ बोल नहीं रहे हैं
सर नहीं बोल रहे हैं हाँ? बोल तो रहे हैं
So what I am going to tell you today regarding what are sericulture and what are the factors which makes this sericulture unique in our country. That is the very important aspects of this seminar. And what I'm going to tell you the main focus of this seminar will be
सर का माइक्रोफोन
the main focus of this seminar will be
Sir, please take your microphone because you are not audible clearly, sir.
yeah. Am I not now?
Yes, sir. Yes, sir.
Hello? Can you hear me now? Hello? Yeah, can you hear me?
Yes, sir. We can hear you.
Yeah. Now we can start. So the science behind the sericulture, endocrinology, and ethnobotany. Now I focus education and employment in India. That is
Sir, you are not audible, sir. I think your microphone is somewhat far
no, I am
away from
connected
you.
with the this my laptop only. Now voice is clear or not? Hello?
Yes, sir. It is clear, but if you can use a headphone or earphone, it will be good, sir. Sir, am I audible to you? सर तो मीटिंग से हट निकल गए हैं
हाँ?
मीटिंग से निकल गए हैं
निकल के फिर ज्वाइन करेंगे
एडमिट कर लो
May I request every participant to kindly mute themselves and turn their video off.
Yeah. Good morning to all.
Yes, sir. You are
Am I
audible
audible
now.
now?
Yes, sir. Yes, sir.
Yeah. Just 1 minute. 1 minute. Yes, sir. Now the core focus of my this talk is the science behind the sericulture and possibility of education. Any pyramid of the sericulture
स्टार्टिंग अगर मतलब ये कंटिन्यूअस अगर
Excuse
बोल
me,
रहे हैं
sir.
तो ये हो जाएगा।
Sericulture. That is Malabar, sir.
ईयरफोन अगर लगा लेते तो होता।
Excuse me, sir.
Yeah. Yes, madam.
Sir, again it is not audible. Can you use headphone or earphone, please?
1 minute. What I'll do is, madam.
Yes.
Why?
Yes, sir.
Because this is showing some difficulty because I'm not audible for this audience. Then I will be not useful. Still, hello, madam.
Yes.
Yeah, madam.
Yes, sir.
Now it's okay. Audible or not?
Yes.
Equally. Okay. Okay.
Be close to mic, sir.
Yeah. I'm close to my madam.
Okay.
Hello. Yes, madam.
Yes,
Yes, sir, you are
Please
audible.
start. Please
Yeah,
start.
there are many varieties of silkworm available in India. That is, as I mentioned in my first slide, that is
Excuse me, sir. We are unable to hear you.
what is technical difficulty? This voice is not clear.
May I request every participant to kindly turn their video off?
किधर से आवाज कम रहा है?
वीडियो आ रहा है, ऑडियो नहीं आ रहा है, नेट कनेक्शन भी सही है, नेट कनेक्शन में प्रॉब्लम होने से वीडियो नहीं आता है।
नहीं, उसमें वीडियो नहीं आएगा फिर।
Hello. Hello.
Yes, sir.
Yes, sir, you are audible now.
Madam, some technical difficulties are because sometimes voice is not. What is it? No. No, can you hear me, madam?
No, sir. We are again unable to hear
Sir,
you.
can you join from mobile phone?
Yeah.
Sir,
Hello?
can you join from mobile phone?
Yeah, I'll connect by mobile only.
Yes, sir. Okay, sir. सबको लिंक भेजना होगा मोबाइल में। इनके पास में है लिंक है, व्हाट्सएप पे भेजा हुआ है ना। इनके लैपटॉप में कुछ प्रॉब्लम है, मतलब ये बोलते जा रहे हैं ना, अगर स्टार्टिंग कुछ रुक रुक करके अगर बोल रहे हैं तो वो उनका माइक्रोफोन रिकॉग्नाइज कर रहा है। कंटिन्यूअस अगर कुछ बोल रहे हैं तो वो रुक जा रहा है।
हो जाता है, ओवर कॉन्फिडेंट हो जाता है।
अभी 11 बजने जा रहा है, सेकंड सेशन हमको शुरू करना है।
मोबाइल से हुआ है अभी, मोबाइल से हुए हैं सर।
साउंड वहां नहीं गया भाई।
Sir, I think that you have joined from mobile, so kindly use the mobile so that we can hear you.
चेक कर, हाँ चेक वही चेक कर।
Sir, on the mobile you will find one option called as connect to audio. So,
Yeah, just one minute. We are checking the system just. I'm sorry for disturbance.
Okay, sir.
Kindly said we have second session from 11 p.m. So kindly
Just 1 minute. Hello.
Yes, sir. Now you are audible.
Hello.
Yes, sir.
Yes, sir. You are audible.
now.
Madam, I'm audible now.
Yes, sir.
Yes, sir. Please start your session.
Yes, madam.
Start your session, sir. We are getting late for second session.
Yes, madam. I'll start. Now, I'll do the.
मोबाइल से यूज नहीं करेंगे, इनका लैपटॉप में कुछ गड़बड़ है, वो बात हो रहा है।
good morning, sir. Actually, there is some issue with our first resource person. He is not audible, sir. So,
And what is very suspected about sericulture, that is whenever the faculty of the fund, not about the faculty of podcast, I can even know how this is so that I have many types of programs, that is assume access and sound. Action access now, first asset of software. And static has global management. Management of the campus and from the forum. Various types of management connected. And appropriate birth prediction is one of the possible and the first aspect for the
सर इसको तो बोल रहे हैं फोन से ज्वाइन करना, फोन से भी ज्वाइन नहीं कर रहे हैं। इन लोग का भी ऐड नहीं कर रहे हैं। Sir, you have joined from this laptop or sir phone? Okay, sir, sir, which name, sir? सर किस नाम से आप ऐड हुए हैं सर? सर अभी तक तो आया नहीं है यहाँ पे। हाँ सर। अच्छा सर वहाँ पे मतलब उस लिंक पे आप क्लिक कर रहे हैं तो ज्वाइनिंग लिखा रहा है क्या? हाँ हाँ। अच्छा मतलब आप ज्वाइन कर गए क्या सर? हाँ तो ज्वाइन विथ वीडियो कर लीजिए सर। आपका वीडियो वहाँ पे दिखा नहीं रहा है सर, या ऊपर में कनेक्टिंग दिखा रहा है? नहीं कनेक्टिंग ऊपर दिख रहा है क्या सर? आप ज्वाइन कर रहे हैं लेकिन हमको लग रहा है किसी दूसरे नाम से आप ज्वाइन किए सर। हाँ सर, हाँ वैसे करके कर लीजिए सर, क्योंकि इसमें फिर आप नाम एंटर कर पाइएगा अपना। नहीं जरूरी है पहले, लेकिन इसीलिए बोले भी थे थोड़ा रिहर्सल कर लेते हैं हम लोग डेमो वाला, तो सबको होना चाहिए वहाँ पे। किस नाम से किया मुझे नहीं समझ में आ रहा। नहीं बोल तो रहे हैं कि हम अपना नाम से किए हैं, लेकिन यहाँ पर मेरे को ऐसा नाम नहीं आ रहा है। नहीं आया है, कौन सा नाम से आया पता नहीं चल रहा। हाँ सर, अब ज्वाइन कर रहे हैं सर। अच्छा तो आप सब ज्वाइन करें तो अभी कुछ चल रहा है ना आपकी स्क्रीन पे, कोई वीडियो चल रहा है कहीं और का? सर आप हमको लग रहा है ज्वाइन कर नहीं पाएंगे सर। वहाँ नहीं सर, वहाँ पे पार्टिसिपेंट्स दिखा रहा है कि कौन कौन पार्टिसिपेंट्स एडेड हैं। अगर वो नहीं दिखा रहा है तो फिर आप ज्वाइन नहीं कर पाए सर। हाँ तो आप स्टार्ट कर दीजिए सर। हाँ स्टार्ट कर दीजिए। नहीं ज्वाइन कर दीजिए सर। सर ये ऋषिका कुमारी के नाम से है सर आपका क्या? दिखा नहीं रहा है आपको तो। पार्टिसिपेंट्स 1 दिखा रहा है। सर हम आपको फिर से एक और लिंक भेज रहे हैं सर, आप उस पर क्लिक करके आप आ जाइए अंदर। हम एक बार फिर से आपको भेज दे रहे हैं सर, व्हाट्सएप पे लिंक भेज दे रहे हैं। हाँ एक और हम लिंक भेज दे रहे हैं, आप उससे ऐड अप हो जाइए। अच्छा सर हम आपको व्हाट्सएप पर में एक लिंक भेज रहे हैं, आप बस उस लिंक पर में क्लिक कर दिए, आप ज्वाइन अप हो जाइएगा। ठीक है सर हम भेज रहे हैं। नहीं उनको सिर्फ लिंक भेजिए, वो हमको लग रहा है दूसरा चीज पे क्लिक कर रहे हैं, तो ये सब पर में क्लिक कर रहे हैं। इनको बस ये वाला लिंक भेजिए, आपको भेज देंगे। खुला हुआ। नहीं कर पा रहे हैं। उनका नाम ही नहीं आ रहा है पर। अब उनको जबकि हम सिर्फ लिंक भेज दिए हैं और कुछ नहीं भेजे हैं, खाली लिंक भेजे। हाँ, खाली लिंक भेजे।
हाँ तो बोलिए ना उनको फोन करके कि आप सिर्फ इस लिंक पर क्लिक करिए।
तो मेल भी कर दे रहे हैं, मेल भी कर दे रहे हैं उनको। अगर लैपटॉप पे खोलना चाह रहे हैं तो मेल कर दे रहे हैं व्हाट्सएप पे। वीडियो नहीं आना चाहिए। नहीं वीडियो नहीं आना चाहिए। हेलो, हाँ सर वो लिंक पे क्लिक किया सर। हाँ वहाँ पर मैं जूम करके आ रहा होगा, जूम या फिर क्रोम लिखा हुआ आ रहा होगा, तो जूम पे आप क्लिक कर दीजिए। हाँ तो जूम। हाँ हाँ सर वहाँ पर मैं ज्वाइन का कुछ ऑप्शन नहीं आ रहा है। हाँ तो ज्वाइन का वीडियो पे क्लिक कर दीजिए सर। हाँ अभी आप आए हैं सर, आ गए सर, आ गए। ठीक है सर, आ गए आप।
मुझे अनम्यूट करिए।
अनम्यूट कर लीजिए मैम।
Hello, sir. Excuse me, sir. Excuse me, sir. Sir, the thing is, it is totally inaudible, and even your PPT is not running, I think. So, sir, kindly join us after one. And in the meantime, you please look after your system, because it is a workshop, and if it is not audible for girls, it is really not useful to them. May I request you to join at 1 o'clock?
Yes, madam. Yes,
Please
madam.
take care of your system and then join us at 1. Thank you very much, sir. We will meanwhile start with the next session, which is about to start at 11. Thank you very much, sir. Please join us at 11. We will be here to hear you. Thank you, sir.
What time, madam?
At 1 o'clock, sir.
Okay,
Please
okay.
join at 1 o'clock. Yes. Thank you, sir. Please look after your system.
Okay.
Thank you, sir. Now we will start second session. Second session will be on the topic endocrinology, and we have with us Dr. N. Venkat Appa Rao. Now I request Ashwarya Bara, Semester 3, Department of Zoology, to introduce our esteemed resource person, Dr. N. Venkat Appa Rao, Assistant Professor, St. Xavier's College, Ranchi. I request Ashwarya Bara, Semester 3, Department of Zoology, to introduce our esteemed resource person.
Can I start?
Yes, Ashwarya, please start.
Good morning, respected Principal, Dr. Sister Jyoti, Head of Zoology Department, Dr. Emma R. Seraphim, and Head of Botany Department, Dr. Anubhuti Singh, teachers of the respective subjects, and my dear friends. I am Ashwarya Bara of Semester 4 of Zoology Department, and it gives me immense pleasure to invite Dr. N. Venkat Appa Rao, Associate Professor of Zoology Department, St. Xavier College, Ranchi, for the next session, Endocrinology. Sir is very popular among students for his unique teaching skill. He has done commendable researches in the field of zoology and has guided many PhDs. We are excited to listen to you and get enriched by your teaching.
Sir, kindly start the session.
Sir, please unmute yourself.
Please unmute yourself.
Sir, kindly unmute yourself.
Am I audible now?
Yes, sir. Please start the session. Thank you, sir.
Okay. My screen is all right?
Yes, sir. Please start.
Means my PPT is visible.
No, sir.
PPT is not visible.
No, sir.
No, sir.
Please share the screen.
So you'll have to share the screen.
Sir, can you see the green colored option at the Zoom share screen?
Yes. Host screen sharing. Okay. Now it is okay.
No, sir.
No, sir,
We
it is
cannot
not
see.
visible.
It's not visible, sir.
Just a minute.
Sir, please click on the share screen option, the green option. And after that, click on the window that you want to share. Or just click on the screen. And after that, click on the share button.
It is all right.
Yes, sir. Now it is visible.
Yes, sir. Yes, sir. Now we can see.
Okay. Thank you very much. The Department of Geology, Nirmala College, Ranchi, and the principal of Nirmala College, Ranchi, for inviting me and providing me this opportunity to share my experience a little bit. Today our topic is endocrinology, I'm N. Venkatappa Rao, Associate Professor, Department of Zoology, Sanjeevis College, Ranchi. In this, we are going to discuss, I'm just giving the broad outline of the topic. First, we'll see what is endocrinology, then what are the glands and how they form, why endocrine glands present in our body, what are hormones, what is the history of endocrinology, why hormones are so specific, and finally we see how do they act. The endocrinology, in fact, a subdiscipline of broader field physiology concerned with the study of endocrine glands, their secretions, functions, and pathophysiology. But to understand the endocrine glands, first we must understand what is a gland in fact. Simply we can define a gland is a cell or tissue which can synthesize a product inside, store it, and release outside. Any cell or tissue, when synthesizing some product, every cell, we know that every cell is synthesizing something inside. But it is not necessary that whatever the cell is synthesizing inside will always release outside. Whenever a cell is synthesizing some product inside, storing it for time being, and as and when required, it is releasing outside, then it is called as a gland. And we also know, you might have studied in earlier classes, that whenever a cell is releasing outside, that is called as a secretory cell. And such secretory cells will have abundant amount of endoplasmic reticulum and Golgi. Until unless these cell organelles are not in abundance, the cell will not be able to secrete properly. And if we consider this cell, majority of the glands contain the cells which are epithelial in nature. However, some glands are also contain modified neurons. Now we say, what is a gland and what type of glands are present? Glands can be categorized in different types. Based on number of cells, the glands can be called or differentiated into unicellular and multicellular. Based on type of secretion, there can be exocrine, endocrine, or heterocrine. Based on mode of release, the glands can be merocrine, apocrine, holocrine, and so on. If we consider a unicellular gland, the entire gland is made up of one cell. Like we can take a common example, that is goblet cell. Majority of the glands which are made up of many cells, like we say gastric glands or salivary glands, these are multicellular glands. If we consider the mode type of secretion, some glands are provided with ducts, like salivary glands, those are called as exocrine glands, and the secretion is always called as enzyme. On the other hand, some glands are without ducts, but these are richly vascularized, and the secretion is directly released into the blood, and the product is called as hormone, like thyroid, adrenal, etc. But some glands are mixed because the part of the gland is provided with duct and part is without duct. Means such gland is secreting both enzymes and hormones. Like for example, we say pancreas. Based on the mode of release of the product, the glands can be merocrine, apocrine, and holocrine glands. In case of merocrine glands, while secreting the product, there is no damage to the cell. Majority of endocrine glands comes under this category. Apocrine gland is that such gland, when there is a release of the product, there is disintegration of some part of the cell, especially in the apical region of the part. And in holocrine, a complete disintegration of the cell is taking place during the release of the product, like for example, sweat glands. Here, through diagrammatically, we can understand how exocrine gland forms, how endocrine gland forms. Then we say the first line, we can see this is this entire lining is representing the epithelial lining. Here, one or two cells are modified, and here these are acting like glands. And here some part of the cells or some layer is enfolded inside. You can see here that enfolded part here is provided with duct and where it is released. And this gland is called as exocrine gland. And here, when the entire gland is formed, it is detached from the male line and it is vascularized. And vascularized gland may be solid, may be hollow. But without duct, then this gland is called as endocrine gland. Here, once again I'll tell, here simply the cells are releasing unicellular or multicellular without any gland. But since releasing outside into the space, therefore it is called as exocrine. These may be enfolded, and that folding may be provided with a space which is called as duct. And into this the secretion is released. This is also exocrine. After the formation, when it is completely detached and blood vessels are innervated and the product is released into the blood vessels, then it is called as endocrine. Such glands can be solid, glands can be hollow or spherical in shape, but never provided with ducts, or the product is never released into the duct. Here, I wish to or try to show that when the product is released outside the cell, there is no damage to the cell. Such a secretion is called as merocrine secretion. And here, in the second case, when the product is released, some part or the apical part of the cell is releasing out. This is apocrine secretion. And here the entire cell is disintegrating. This is called as holocrine. These three types of secretory process can shown by the cells. Majority of the endocrine glands comes under the category of merocrine nature. Some these milk secreting glands, these are apocrine in nature, and some sweat glands are, we have holocrine in nature. Now, I said also that not only epithelial cells are forming the gland, sometimes modified neurons also responsible for the formation of gland. These are called as neurosecretory cells. Here I tried to show the normal neuron and here secretory neuron. And normal neuron, we say, we know that having a cell body or cyton surrounded by the dendrons, a long axon, and the neurotransmitter is released into the synaptic region. But here, this cell is modified, here also having dendrites or soma or cyton, axon, but in the terminal region, instead of synaptic area, there present capillaries where the secretory materials are releasing. This is called as a modified neuron or neurosecretory cells. But since it is directly released into the blood capillary, therefore this is also hormone, but more specifically, this is called as neurohormone. So gland may contain epithelial cells, majority of the gland, but it is not necessary that every gland should contain epithelial cell. Certain glands may also contain a modified neuron. The common example is neurohypophysis. So far what we can understand that the endocrine glands are ductless glands which secrete their product directly into the blood, and the product is called as hormone. Majority of endocrine glands contain or are epithelial in nature. However, some can be more of modified neurons or neurosecretory cells. Just I have given the example that the example of the neurosecretory cell containing endocrine gland most common is neurohypophysis of pituitary. Now, up to the stage, I wish that whether any student having any question, then the students are free to ask.
ठीक है सर.
Okay. अच्छा. Okay.
Hello. Hello.
Okay, so those who want to ask some question, they can ask now.
Yes, they can ask now. At this point, if they want to ask some question, they can ask. You can unmute them. They are free to ask the question so far, what I have said.
Question will be filtered. Unmuted. Unmuted.
Students, you are muted. If anybody has any question, please ask now.
Up to this, anybody having any problem in understanding so far I said, they can ask. Because I have, for your convenience, I have divided into topics that what is gland, what is endocrine gland. So far I said.
Sir. Good morning, sir.
Good morning.
Sir, so you said that some glands secrete epithelial cells, but if someone asks
some
you
gland,
ठीक है ना
please. Go on, go on.
Am I audible, sir?
Sure, you are.
So why it is said that some of them does not contain epithelial cells?
See, I said earlier, what is a gland? Any cell which is capable to synthesize inside something and release outside, it is considered as a gland. Clear?
Yes, sir.
Okay. Now, what type of cell it can be? So, whenever we say that a cell is secretory cell, at tissue level, we see that it is epithelial cell. Because you might have studied about the tissue, epithelial tissue, connective tissue, muscle tissue and nervous tissue.
Yes, sir.
Okay. So, the joining tissue, not secretory. The muscle tissue is biological motor tissue, responsible for movement, not secretory. The nervous tissue is responsible for coordination and communication. So therefore, the tissue left behind for secretion is epithelial tissue.
Okay,
Clear?
sir. Yes, sir.
You have any doubt, then feel free to ask.
No, sir, I'm clear.
Therefore, we see in majority, 99% of the glands contain epithelial tissue. Because epithelial tissue is responsible for protection. Epithelial tissue responsible for secretion. Okay?
Yes, sir. It is clear. Thank you, sir.
So, whenever a secretory cell is present, normally it is epithelial tissue. And 99% of the glands, therefore, or secretory tissue is epithelial in nature.
So, sir, we can say simply that because of its nature, that it is secretory, that's why it is present more in glands.
Then it is, if we say that clear, see, your name, may I know your
So,
name,
my
please?
name is Neha.
Neha.
Yes,
Neha, whenever, see, you fix it, whenever we say a gland, it contains epithelial tissue. Because epithelial tissue having the property of secretion.
Okay, sir.
Sure, okay?
Yes, sir.
Any tissue involved in secretion, that is always epithelial tissue.
Okay.
Therefore, a gland, if it is secreting something, it should contain or it will contain epithelial tissue.
Okay, sir.
Clear?
Yes, sir. Now I'm clear.
Now,
Thank you, sir.
the second is that now we say if a gland is epithelial tissue, 100% of the glands are made up of epithelial tissue. The question mark is coming. Then we say there are glands, example, which are not of epithelial tissue but contain modified neurons.
Okay, sir.
Okay. So
Yes,
what
sir.
can be, what type of cells can present in gland? A gland contain, majority of gland contain epithelial tissue. However, some glands may have modified neurons. If a neuron is secreting that is called as neurosecretory cell.
Okay, sir.
Am I clear naha?
Yes, sir. You are clear to me. Thank you, sir.
Now, to fix up in your mind this concept wise, whenever a gland, whether it may be any exocrine gland or endocrine gland or mixed gland, it will contain epithelial tissue, or very few are there which contain modified neurons or neurosecretory cells.
Okay, sir.
Okay?
Yes, sir.
Anyone having other questions? Can I? Hello?
Yes, sir. There are no questions. You can proceed, sir.
Can I? Okay, thank you. Now, the study is coming. The next is why the endocrine glands are existing in our body. Now, students, you might have studied the taxonomy in your +2 level or in your UG level. That first unicellular came, then unicellular converted into or evolved into multicellular, and then after that tissues came, that after that organ, and then system grade of organization existed. That's how we understand the evolution of the animals. So here we see a multicellular organism when existed in the universe, and with the evolution of tissues, organs and system grade of organization, there arose a need of coordination and communication between tissues, organs and systems for the proper maintenance of life. To meet this demand of coordination and communication, further two systems are evolved. One is called as nervous system, which include neurons, and other is called as chemical system, which include hormones. Now, how we can understand this? See, you might have studied about an amoeba. For nutrition, one amoeba need not to tell other. For respiration, one paramecium need not to communicate to other. It is self-dependent, single individual made up of one cell. Such animals, we say, those are protozoans. But when we move towards the tissue grade of organization, like you might have studied about the coelenterids, where the tissue grade of organization first came into existence. Then we have helminths onward, we have organs, and then arthropods onwards, we have different systems are there. If we consider our body, take an example, our body does not contain a cell, but rather large number of cells which are united to form tissue, and such tissues are present in an organ, and such organs are united to form system. I'll give you an example of one digestive system which having the alimentary canal. See, we have alimentary canal, we simply say that the alimentary canal is responsible for the process of digestion after taking the food. But you might have studied in your earlier classes that the alimentary canal is taking utmost care for the proper digestion. For that, it is involved in coordination, communication. Like we consider say, for example, in case of stomach, when we are swallowing the food is entering into stomach. What is the function of stomach? The function of stomach is two. Number one is peristalsis, and number two is digestion. Both must be in a coordinated way. If as soon the food is reaching, the muscles are showing peristalsis, the food will not stay in stomach, rather it will move forward without any digestion. Therefore, as the food reaches, first there is a secretion from the gastric glands so that the food can be digested. Once the digestion has taken place, then only there is a peristalsis of stomach so that the chyme can move forward. Means there must be coordination between the movement of alimentary canal and the secretion of alimentary canal. And you might have studied For the movement of alimentary canal, nervous system is also responsible. For the secretion of alimentary canal, the nervous system is also responsible. For the movement of alimentary canal, the hormones are also responsible, and the secretion of alimentary canal also requires the hormones. Together, the nervous system and the hormonal system responsible for the proper functioning of the stomach. Like you might have studied, that between longitudinal and circular muscles of the stomach, there present Auerbach plexus, which control the movement of alimentary canal. And in the submucosal region, there present mucinous plexus, which is responsible for controlling the secretion of alimentary canal. Similarly, in the wall of inner surface of stomach, you have gastrin, hormone secreting cells, which regulate the secretion of gastric juice. You have the 5-hydroxytryptamine secreting cells, which is a powerful stimulant of gastric peristalsis. Here, both nervous system and the hormonal system together responsible for proper coordination and communication between the tissues of stomach, so that the organ stomach, one organ that is stomach, can function properly. Such actions we can observe in different parts of the alimentary canal. Then only there is a proper digestion of food. So that's why we can easily understand that whenever organ grade, tissue grade, system grade of organization we can see, there is a need of coordination between and communication between tissues, organs and systems. To meet the coordination communication, there is nervous system and the chemical system. That's why you might have studied the nervous system for the first time we can see in coelentrates, because in coelentrates we can see the tissues first. If we compare these two systems, the nervous system and the chemical system, the nervous system is fast acting, approximately 300 meters per second in our body. And the hormonal system, a little comparatively slow. It requires 1 minute to reach from one part of the body to other. So finally, what we say, the endocrine system or endocrine glands are responsible for coordination and communication between organs, between systems, and between tissues for the proper maintenance of life. Now, we consider what are hormones. Literally, the hormone means to excite or to arouse. Means to stimulate, to provoke, to evoke. That is the literal meaning of the hormone. But how we define hormone, the hormones are called chemical messengers, or better we say chemical modulators, secreted by endocrine glands, released into the blood and reach to the target tissue, which is normally away from the secretory tissue, thereby modulate the activity of the target tissue. Once again, I'll say, hormones are generally or commonly we say chemical messengers, but better these should be called as chemical modulators, secreted by endocrine glands, which are ductless, therefore directly released into the blood. Through blood, these reach to the target tissue, and the target tissue is normally away from the endocrine tissue and modulate the activity of the target tissue. Why I said a modulator? Because it is not only stimulate, the target tissue, it can, acting target tissue can be inhibited. A molecule which can stimulate or inhibit, together if we consider that function, then we say it is a modulator. That's why nowadays the hormones are called as chemical modulators. Usually we find that the endocrine tissue and the target tissue is distant, little away from each other. But some hormones are there which release in the close proximity of the endocrine tissue. Such hormones are, we refer as local hormone. When we say local hormone, then only we can understand that it is released in a close proximity. But normally, when we say hormone, it is reaching to a little far from the endocrine tissue to the target tissue. I have given one or two examples of this local hormone like prostaglandins, kinins. These are the examples of local hormones. Now, but when we say FSH, LH, those are normally called as hormones. Up to this, any question from any student? Please once again unmute and then ask the students so that they can ask any question they have.
Students, you all are unmuted. You may ask question. If there is any, you may ask question to sir.
No questions?
Good morning, sir.
Good morning.
Sir, what is the function of prostaglandins?
Prostaglandin, see, the prostaglandin, first of all, why the name is prostaglandin is given. The prostaglandin, first of all, been observed to be secreted by the prostate gland of male.
Yes, sir.
Okay, in male reproductive system, a prostate gland is present. In that, first of all, in the secretion, first of all observed, that's why it is called as prostaglandin.
Yes,
But
sir.
now it is being observed that it can be secreted from almost all tissues of our body.
Yes, sir.
And whenever there is a secretion of prostaglandin, we always feel pain.
Pain.
Pain. दर्द जिसको बोलते हैं हिंदी में.
Yes, sir.
Okay.
Yes, sir.
How this prostaglandin, say for example, you are pressing one tissue.
Yes, sir.
You can see me, I am pressing my hand.
Yes, sir.
So you can feel pain because there is a secretion of prostaglandin and that prostaglandin affects the algesic receptor.
Algesic.
Which algesic receptor, ALGESIC, algesic receptor.
Okay, sir.
Okay, that is taking that message to our central nervous system, then we have the feeling pain.
Yes, sir.
I hope Divya is asking.
Yes, sir.
Okay, Divya. Now, Divya, you might have taken painkiller.
Yes, sir.
And what is written in that painkiller? It is normally written as analgesic.
Yes, sir. Yes, sir.
An means no, algesic means pain.
Yes, sir.
Okay.
Hmm.
Any analgesic is there, it always inhibits the secretion of prostaglandin. That's why you are getting relief from the pain.
Pain. Yes.
Okay.
Yes,
But
sir.
it is not that somewhere you have a cut and the other place you have a pain.
Yes.
Okay. This is a local hormone. The tissue which is secreting prostaglandin, in that area only you will feel pain.
Yes, sir.
Okay, whatever analgesic you are taking, that is the inhibitor of prostaglandin, thereby you are getting pain.
Relief from the pain. Yes, sir. Thank you, sir.
Right?
Yes, thank you, sir.
So next I'll tell you what are kinins.
Yes, sir.
Whenever there is a cut on the body,
Yes, sir.
the blood vessel will constrict to minimize the loss of blood.
Yes, sir.
The constriction of blood vessel is again due to the secretion of kinins.
Okay, sir.
Okay, these are local secretion is there and always affecting the local tissues.
Yes, sir.
Clear?
Yes, sir. Thank you, sir.
Anyone more? Any question more? Can I move further?
Yes, sir. Please.
Okay. Now, if we consider in our body, there present large number of endocrine glands. We are enlisting some of these like pineal gland, pituitary gland, hypothalamus, thyroid, parathyroid, thymus, pancreas, gonads, like testes, ovary, then placenta. These all are considered as endocrine glands, and sometimes we say altogether this is known as endocrine system. But according to today's knowledge, each and every cell our body can act an endocrine cell. That's why at least it can secrete prostaglandin. This underlying diagram only have showing some major endocrine glands. Here in that, it is not also shown that alimentary canal, which is the largest endocrine gland. Okay. Now we say that in our body, a large number of endocrine glands are present. And these endocrine glands are secreting, each and every endocrine gland secreting hormone. And all hormones. Now, how this endocrinology study has started? This is the first recorded history of endocrinology. The experiment of Berthold in 1849. In his experiment, he has taken 4 groups of cockerels. He has seen that in the first group, there was no surgery, and that is called as a normal group. And he observed that in the cocks, the combs are well developed, and these cocks show interest towards the hen, and the aggression fighting behavior is observed, and a large croaking sound of the normal cock is there, we all know. In the second group, what he did, from the cocks, he has removed the testes. Then he observed that the comb was poorly developed, and the cock does not show any interest in the hen. The sound was, or the croaking sound was very less, and they are always sublimed and never show a fighting behavior or less fighting behavior. In the third group, he has taken the testis, one testis. We know that the cock contains pair of testes. He surgically removed out one testis. Then what he observed, the remaining testis is increased in size, and the cocks show normal behavior. In the fourth group, he has transplanted the testis from one to other. Reciprocal transplantation has taken place of the testis. Even then, the testis here also grown larger in size, one testis, and the cocks show a normal behavior. Then Berthold has concluded that there is something is secreted from the testis, because without testis, there is, here we can see that the first group, it is normal. The first group is normal, and the second group is both the testes are removed, and in the third group, one is removed, and the fourth group, cross-transplantation has taken place. So when there is no testis, the cock does not show its normal property. One testis is there, it is grown in size and showing the normal property. In cross-transplantation also, the testis increased in size and showing the normal property. Then Berthold concluded that there is something secreted from the testis which is responsible for the characteristic of cock. Now we say that the secretion from the testis is testosterone, which is responsible for the masculine behavior of the cock. When the testis is removed, the masculine behavior is perished, and when one testis is there, it is increased in size. Nowadays, this is called as compensatory hypertrophy. When instead of one, instead of two, when one organ is present, that one organ is increased in size to compensate the functioning of other. That is called as compensatory hypertrophy. This compensatory hypertrophy can be seen in third and fourth row. This is the first recorded history of endocrinology when Berthold shown that testis is responsible for the secretion. He never explained that what is it, and it is whether it is a hormone, whether it is something else, he has not explained, but he has shown that the testis secretion is essential for the masculine behavior in case of Cox. It is in 1849. After 1849, about 50 years later, only the endocrinological research got impetus with the discovery of secretin by Balsis and Starling in 1902 and the hormone term coined by Starling in 1905. After 1849, we are directly coming to 1902 when Balsis and Starling discovered the secretin. They have seen that the duodenal extract when injected responsible for causing the secretion of pancreas. That's why they have given the name secretin. They observed this in 1902 and in a conference, the Starling coined the term hormone for the secretion in 1905. Then after the branch of endocrinology grown with rapid pace, and nowadays if you see, the molecular endocrinology is the need of the hour. Everything in endocrinology also, we are not leaving the molecular biology. Molecular endocrinology is not now the need of the hour, and majority of the workers now are working or doing the research in molecular endocrinology. So what we can say so far? So far we can say that the endocrine glands secrete hormone into the blood, reach the target tissue, thereby modulate the activity of the target tissue. And this hormone term was coined by Starling. Although in 1849, Berthold has given some historical record for the functioning of the gonad or testis, the secretion of the testis in the masculine behavior in case of Cox, but really it is started in 1902 with the discovery of secretin by Balsis and Starling and the hormone term coined by Starling in 1905. Just we have seen that our body contains large number of endocrine glands, and each one is releasing number of hormones, one or many hormones, and all hormones are released into the blood, and we know that the blood is circulating all over the body. Therefore, we can easily understand that at a time within blood, large number of hormones are present. Surprisingly, each hormone affecting a specific target tissue only. Once again, I am repeating this. The history of endocrinology we have seen started in 1849 with Berthold, but only after about 50 years the Starling and Balsis work came into existence. Then the real endocrinology has started, and what we know today that the endocrine glands are secreting hormones into the blood, but the term hormone is coined by Starling. The first hormone was secretin, and these hormones are reaching to the target tissue and modulate the activity of the target tissue. Just we have in earlier slide, here we have seen that in our body large number of endocrine glands are present, and each endocrine gland is richly vascularized. Each endocrine gland is secreting large number, one or many hormones, and all hormones are released into the blood, and the blood is circulating all over the body. Means at a time within blood, there will be large number of hormones are present, but we said that hormone is specific to target tissue. When we say hormones are specific to target tissue, is it correct? If yes, then hormones must recognize the target tissue and act on it. Again, I request to the organizers to ask the students that any confusion up to this stage.
Yes, sir. If students are unmuted, they can ask the question. And sir, I request you that from 12 we will have the another session.
Yes. I'll try to complete.
Yes. Thank you, sir.
Because just I'm giving one or two question possibility to the students that they can feel comfortable.
Yes. Okay, sir.
Any question, Divya, Neha? Unmute yourself.
No, sir. We understand properly.
Okay. Can I move forward?
Yes, sir.
Yes, sir.
Okay. Now, see again, I'll tell you, we say hormones are specific to target tissue. Is it correct to say? If yes, it means hormones are recognizing the target tissue and acting on it. But it is not true. In fact, the target tissue is recognizing the hormone and respond accordingly. Once again, I'll say, the hormones, we say that acting specifically on target tissue, but it is not true. In fact, the target tissue is recognizing the hormone and responding accordingly. For this, each target tissue having specific receptor to recognize a hormone. Like, for example, if we say FSH acts on ovary, it is not due to the FSH, rather due to the receptor of FSH present in the ovarian cell, thereby it responds. Okay? It is not the property of FSH that it is acting on ovarian cell. It is the ovarian cell which having the receptor, thereby it is recognizing FSH and then responding to FSH. Now the second is that when we say hormone, these are chemical messengers or chemical modulators. What chemistry is behind it? We say the hormones basically can be of three types. Number one, called as peptides or proteins, which are polymer of amino acid residues. You might have studied in your earlier classes. The second is steroids. Steroids are nothing but derived lipids. And third is modified amino acids. Sometimes one or two amino acids are modified and act as a hormone. These three categories of hormones are present. One may be peptide or proteins, which is a large polymer of amino acid residues. Second is steroids, which are lipid derivatives. And third is modified amino acid, where one or two amino acid, like single tyrosine molecule, can convert into or modified to act as hormone. Such hormones are also present. Now we say hormones are acting on target tissue because the target tissue having receptors. Now here I have given the list that where the receptors can locate. Just I show you through this diagram first, that the receptors, if this is a target cell, the receptor can present on cell membrane, that is called as membrane receptor. It can present in cytosol, that is called as cytosolic receptor. It can present within the nucleus, can be called as nucleoplasmic receptor. It is not that receptor in a target tissue present in one location. Receptor may present on cell membrane, receptor may present in cytosol, receptor may present in nucleoplasm. Okay? But whenever we say receptor, it is a protein. And it may be multi-unit protein, different, more than one molecules are united and then acting as a receptor. Or if one molecule, then it can be a multi-domain, means many areas are there for this protein. In next we can understand. So here I have shown that one, that the receptors are present on the cell surface, may present on the cell surface or present inside the cell. If cell surface, the receptor may be tyrosine kinase linked. group A linked to TK. Here TK stands for tyrosine kinase, and it may be linked to G protein, guanosine coupled protein. If tyrosine kinase, it may be intrinsic or may be extrinsic. If tyrosine kinase, it may be adenylate cyclase or phospholipase. Superfamily 2, once inside the cell, it can be cytosolic, it can be nuclear. Just as shown. This will understand in next 1 or 2 slides. Here I have shown that it can be present on cell membrane, can be present in cytosol, can be present in the nuclear plasm. Here just I have shown that you might have studied about the cell membrane. How the membrane receptors are there. Membrane, we have studied in our earlier classes that it having bilayer of lipid where proteins are arranged in mosaic fashion. Some of the proteins are unitedly acting like receptor. Here I have shown that these 3 unitedly acting like receptors. This is one molecule, this is another, and this is third. This is called as recognition unit, which is always to ectodomain of the cell membrane, outer surface of the cell membrane, and this is transduction unit across the membrane, and this is catalytic unit towards inner domain or endodomain of the cell membrane. Once again, I'll tell you, when we say receptor present on the cell membrane, this cell membrane receptor may be multi-unit protein. I have just told that it may be made up of more than one protein together acting like, here I have shown, receptor may be made up of 3 protein units. One unit is called as recognition unit, which is towards outside. Another is transduction unit across membrane, and third is catalytic unit, which is towards inside. These together are unitedly called as one receptor. To this recognition unit only hormone attaches, then it is passing the message to transduction, then to catalytic unit. This together called as one receptor. That's why I said one receptor can be a multi-unit protein. This is a simple example of a adenylate cyclase system receptor. Another example is tyrosine kinase system receptor. So here we have seen that these proteins are present in the form of 2 outside and 2 inside. These proteins are called as alpha proteins and beta proteins. Means this is entire is one receptor comprising 2 alpha units and 2 beta units. This beta unit is towards inside the cell membrane and alpha unit is towards outside the cell membrane. Whenever attaches to the target cell, it is towards from outside to alpha unit, it will go inside through beta unit, which can get tyrosine activity. So here I have shown the receptor also present inside the cytosol, inside the nucleus. So cytosolic receptor we can see here one domain. This is called as domain, and this is another called as domain. This domain is hormone binding domain, and this domain is DNA binding domain. That's why I said these are the receptor may have multi domains. Here also having hormone binding domain and DNA binding domain, because all these 2 receptors are always acting via nuclear transcription, means act at the level of DNA. The plasma receptors, plasma membrane or cell membrane receptors always send the signal through enzyme, maybe adenylate cyclase or tyrosine kinase. But the cytoplasmic receptor and the nuclear receptors are acting through DNA. Here I have shown one example of the membrane receptor. Here, this part is, this is known as the recognition unit, which is C. This is hormone binding area which is exposed out. This is transduction unit. This is again multimer, alpha, beta and gamma, and this is catalytic unit. This is, I said, adenylate cyclase. So here the together this one, this recognition unit, this transduction unit and the catalytic unit, these together called as one receptor. When the recognition unit, with the recognition unit, hormone binds, then only the recognition unit comes in association with transduction unit. You can see here the transduction unit having alpha, beta, gamma. This alpha is present in low energy state. That's why it is called as GDP state, diphosphate state. Once the hormone is attached, this recognition unit attached with the transduction unit, the alpha subunit of the transduction unit will get phosphorylated. Means this addition of GTP will be there. The GTP will be added to this. When GTP will be added, then the alpha unit get phosphorylated. The phosphorylated alpha unit get detached from beta and gamma. That beta and gamma will separate out. Then the alpha unit will attach with catalytic unit, that is adenylate cyclase. And then when attaches with this catalytic unit, then only the active site of adenylate cyclase will form, and then the ATP will convert into cyclic adenosine monophosphate. The enzyme which converts ATP adenosine triphosphate to cyclic adenosine monophosphate, this enzyme is called as adenylate cyclase. And this cyclic AMP now takes the message inside the cell. Once again, I'll tell you, any membrane, cell membrane, just we have seen here, the cell membrane receptor is there. This membrane receptor is responsible for recognizing the hormone, and only the message will go inside the target cell. After taking the hormonal message from outside, inside the cell, another messenger will be produced, that is called as second messenger. When the membrane is present on the cell membrane, when the receptor is present on the cell membrane. To explain that, we have seen here that receptor which is present on the cell membrane will have three units. One is recognition, second is transduction, and third is catalytic. Recognition for hormone binding, transduction for message propagation, and then catalytic unit to produce messenger, second messenger. Then we have shown here, this is the recognition unit, this is transduction unit, and this is catalytic unit. Catalytic unit is always an enzyme. The recognition unit, with this recognition unit, the hormone binds. When recognition unit with the recognition unit hormone binds, the recognition unit undergo change and binds with the transduction unit. The transduction unit earlier present in low energy state, GDP. When recognition unit binds with the transduction unit, then it will be energized with the help of GTP. Since it is energized with the help of GTP, that's why it is called a G protein. Once energizes the transduction unit, which comprises alpha, beta and gamma, the alpha gets separated from beta and gamma and get attached with the catalytic unit. Once it attaches with the catalytic unit, the enzyme now activates, the active site will form. With the active site, the ATP will bind. The ATP binding causes the formation of cyclic adenosine monophosphate. This is the second messenger. This will carry the message inside the cell. This is how the membrane receptor, which is G-coupled one, that will act. Question from the students. Organizers, please unmute the students.
मैम ये ऐड हो गई है मैम
Students, you are unmuted. You can ask the questions.
Hello.
Hello, students. You can ask
Excuse
the
me, sir.
question.
Yes.
Sir, once again, multi-domain protein, sir.
See,
Yes, sir.
protein, if we consider, you might have studied in your semester 3 and 4. The protein present in different structural form.
Yes, sir.
Secondary, tertiary, and quaternary. To attain a functional status, the protein must present in a tertiary form. Okay?
Yes, sir.
Khushi Kumar. Okay.
Yes, sir.
And the tertiary form of protein will have many folds.
Yes, sir.
And the folds are in fact the functional areas of the protein. Okay?
Yes, sir.
You understand this line or not?
Yes, sir.
Say for example, one enzyme is there. If it is a functional molecule, it should have many grooves. One groove where the substrate attaches, other groove the cofactor may attach or coenzyme may attach. You understand?
Yes, sir.
Then only it can act. Like here, receptor is also a functional protein. It must attach with the hormone and it must execute the functioning of the hormone. Therefore, a protein molecule have many folds.
Yes, sir.
Here,
Yes, sir.
each fold is known as a domain. Here you can see here, I have shown in this diagram that this is a protein molecule which we consider as a receptor. Here this having a domain where hormone can bind. Until unless the fold is not there, the hormone will not bind. And this is the fold where it will go and bind with the DNA. This is called as hormone binding domain. Means area, means a fold where something attaches.
Okay, sir.
Okay, so here also we can see that the recognition unit having a domain where hormone binds.
Yes, sir.
Okay, another domain attaches with the transaction site. The GTP, when convert into GTP alpha, convert into GTP alpha, it is and attaches with another domain of catalytic site. Domain means area or fold, where one molecule can attach into the protein.
Yes, sir.
Okay, a functional protein will have many such areas are present.
Okay, sir.
That's why we say it is a multi-domain protein.
Okay, sir. Thank you.
Domain is nothing but a fold, an area where a molecule can bind.
Okay, sir.
Any question more? Madam?
Sir, I think it is. Yes, sir. Please, sir. Please. Because next resource person has entered. She is waiting, sir.
Yes.
Okay, sir. Thank you, sir.
Thank you all for your patient hearing. I am thankful to the principal of Nirmala College, Ranchi, and the faculty members of the Department of Geology, Emma Madam, Anjali, Smita, and Saraswati. We say Veena Pani. Thank you all.
Thank you so much, sir. Thank you so much.
Thank you very much, sir. Thank you. We are always happy to be with you. Thank you very much, sir.
Thank you so much, sir.
I have more slides are there, but I think it will be.
It's all right, sir.
Thank you, sir.
Thank you very much.
Thank you so much, sir. Thank you, sir.
Over to Nikki. Nikki, please start the third session.
Good afternoon, everyone. I, Khushi, on behalf of our department, Zoology and Botany, would like to extend a hearty vote of thanks to you, dear sir, for gracing to you our important work and sharing with us. A very big thank you for your precious time you have given us in your own busy schedule. We are very fortunate to have you as our mentor, sir. I'm running short of words to express our gratitude towards you. Both, but I am sure that we will make your efforts true. Keep sharing your blessings upon us, sir. Thank you once again.
Thank you. Thank you very much.
Thank you, sir.
So we will start next session, which is on the topic Ethnobotany. And we have with us Dr. Purvi Saikia. Now I request Ruthvi Prasad, Semester 3, Department of Botany, to introduce our esteemed resource person, Dr. Purvi Saikia, Assistant Professor, Central University of Jharkhand, Ranchi.
Thank you, Nikki. Good afternoon to our respected Principal, Dr. Sister Jyoti, Vice Principal, Dr. Sister Shobha, our resource person, Dr. Purvi Saikia, all the teachers and students present in this workshop. This is my honor to introduce Dr. Purvi Saikia ma'am. Dr. Saikia ma'am is an ecologist who did her PhD from Merit State University. Her expertise is in plant ecology, biodiversity conservation, soil sciences, plant taxonomy, agronomy, forestry, conservation of endangered species, as well as medicinal plants. She has 8 years of experience and she has published about 19 research papers in different national and international reviews. She is a recipient of research grant from SERV, GOI and the Young Scientist Scheme, UGC Startup Grant and more. Earlier she was working as JRF and SRF. Presently she is working as Assistant Professor at Central University of Jharkhand, Ranchi. So on behalf of entire fraternity of Nirmala College, I Ruthvi Prasad, student of Nirmala College, welcome Dr. Purvi Saikia ma'am for her presentation on the topic Ethnomedical Plants of Jharkhand. Thank you ma'am, you can continue.
Thank you, Ruthvi.
Thank you.
So before going to start, I just want to congratulate the entire Nirmala College fraternity for conducting this online workshop. So as I am already 5 minutes delay, so that's why without any further delay, I just want to start my presentation. And today I am going to talk about the ethnomedicinal plants of Jharkhand, with special reference to immune booster plants to fight against COVID-19 pandemic. So before going to directly to the ethnomedicinal plants, so just I want to give a brief overview about the COVID-19 pandemic. As we all know, the entire globe is suffering the severe health concern due to this COVID-19 pandemic, and this COVID-19 is caused by the coronavirus SARS-CoV-2. SARS is severe acute respiratory syndrome, coronavirus 2. And this disease has been originated in Wuhan in the central Hubei province of China. And the first case of coronavirus was detected on 31st December 2019 in China, and later on in Thailand in January 2020. And most of us are aware, means what is the suspected origin of this COVID-19, and suspected bat is the origin of COVID-19. But recently in current biology there is one article published probable pangolin origin of SARS-CoV-2 associated with the COVID-19 outbreak. So recent research confirmed not only the bat, pangolin is also a suspected origin of this SARS-CoV-2. And as we all know, it has already spread, this outbreak has been spread over 215 countries, and this COVID-19 is called as a zoonotic disease. Why? Because it occurs at the interface between humans, animals, and the environment. And as we all know, WHO declared it as a public health emergency of international concern on 30th January 2020. And in February 2020, this epidemic turns into pandemic due to its human-to-human transmission and its worldwide spread. So, this is the recent report. Here you can see already almost 25 million people have been affected by this COVID-19 pandemic. But the good thing is that recovery rate is quite high. That is almost 17 million people throughout the globe. So just I want to share the severity of the COVID-19. This is a map which I have been published recently in Science of the Total Environment. So it's a publication of the April. And here you can see India is in light orange color. That is, we had during April 10th of April 2020, we had less than 10,000 cases. And USA was in the top position with highest global cases, followed by Spain, Italy, Germany, France, China, Iran, and EU. And see the present scenario. In the previous slide, you can see India was nowhere in the scene. So now we are holding the third rank in terms of infected cases, COVID-19 infected cases. USA in the top rank, followed by Brazil and then India. And in India, the first case was reported on 30th January 2020, where the first case the first COVID-19 cases has been reported by 31st December 2019. After 1 month, the first case has been found in India, that is in Kerala. One student came from China. And later on, a set of cases was recorded across Delhi with a tourist group of 14 Italian, 1 Indian, and that is on 3rd March. Means after 1 month, almost 1 month, you can see from 1 case to it has been enhanced like 15 cases. And the major outbreak of COVID-19 started in India by March 2020. And here you can see that it is a global scenario. Here you can see by this red line, I am trying to show the trend of the increasing global cases. You see, from starting to till that, it's increasing day by day. And the best part is that the recovery rate, is by August, it is crossing the rate of infection. Means average recovery is more as compared to the average infected cases of the worldwide. And you can hear by this gray line, you can see like the total mortality and that toll, and it is almost in a straight line. Means it's not increasing. That is the best part in COVID-19. And in this second graph, I will shift this one. In this second graph, you can see that global coronavirus cases, it's rising so fast. Because as we all know, because coronavirus is transmitted from human to human. At the initial level, we had less cases. That's why the rate of infection was also less. And presently, the case is enhancing. That's why the rate of infection is also enhancing. You see, from 5 to 10 million cases, moving from 5 to 10 million cases, it took 38 days. Where from 15 to 20 million, it takes only 20 days. So obviously, when the cases are rising, obviously the rate of infection is also rising. And here you see the top countries with daily new cases. So India is in the top with almost 80,000 cases per day. While USA in the second rank, Brazil in the third rank, with as compared to India, they have very minimum cases, daily cases. And presently, we have 34.5 lakhs infected cases, and out of that, recovery rate is 76.5. So recovery rate is quite high. And if we see the death rate per 1 lakh people, you see it's range from in other countries 61.8 to minimum of 49.6, while in India the death rate is very low, that is only 4.5. So today I am here not to talk about COVID-19 pandemic, but I am here to talk about the ethnomedicinal plants of Jharkhand. As I am working in the field of forest ecology, so I have already completed 2 R&D grants on the projects, R&D projects on the forest of Jharkhand. As we all know, Jharkhand is one of the most biodiversity rich country of the India with extensive forest resources with 29.62%, that is higher than the national average. And why? Because of its origin, physiographic as well as climatic condition. This is the map I have taken from the FSI report 2019. Here you can see in our Jharkhand, we have maxima. This deep green color is showing like very dense forest, light green color is showing moderately dense forest, and yellow color is showing like open forest. And here you can see most of the very dense forests are located in Lohardaga, Latehar, Chatra, as well as in West Singhbhum district. So the Jharkhand is located mainly at the Chotanagpur Plateau region, and this Chotanagpur Plateau is formed a link between Satpura hill range and the eastern Himalaya that allowed species exchange. So that's why Jharkhand has a very huge forest diversity. And in this second map, I am just trying to show the all 24 districts of the Jharkhand. And this is a photograph taken by my student during the field visit to the Silli forest range of Ranchi. I'm just trying to show the overview of the forest of Jharkhand. And the state, as we all know, Jharkhand, the name itself signifies that it's rich in forest resources. Jhar means forest, Khand means land, land of forest. The state has luxuriant forest vegetation with magnificent undulating hills as well as valleys. And the forests of Jharkhand are mainly tropical deciduous type, and sal forest is the most dominant vegetation. Here you can see in this graph, in this table, like Jharkhand has the highest amount of dry peninsular side forest, that is almost 53.77%, followed by northern dry mixed deciduous forest, that is almost 35%. Besides that, in Jharkhand, we have moist peninsular low-level salt. This classification is actually based on the Champagne and Sake 1968 classification taken from the FSI report 2019. So in Jharkhand, we have also moist peninsular berry salt, dry deciduous scrubland, we have Rogelia forest, we have Butea forest, dry bamboo brake, as well as plantations, which is considered as tree outside the forest. And as I mentioned, sal forest is the most predominant vegetation of Jharkhand, and the dominant associate of Sporia robusta is Diosporus granogynum, Terminalia species. In Terminalia, we have so many species in our forests like Terminalia cibula, Terminalia valerica, Terminalia ortuna, Terminalia tomentosa, and so on. Similarly, as it is a dry deciduous forest, so we have association of acacia species, we have Moduca longifolia, we have Busnenia cochinensis, then Butea monosperma. And here you can see like forest cover change matrix of Jharkhand on the basis of FSI 2017 and 19 report. And here is a positive change, like this is non-forested area, 77 hectare area has been changed to very dense forest, moderately dense forest, open forest, and scrubland. And maximum modification has been taken in terms of open forest. So the main reason for the increase in forest cover in Jharkhand are mainly due to the plantation as well as conservation management and improvement in the interpretation, because this coverage of forest is used to be calculated based on the satellite imagery. So now I'm coming to the wild plants of Jharkhand. So as we all know, India is a tropical country and it is considered one of the 17 mega biodiversity nations. So India has approximately 800 edible plant species that are grown in wild and consumed mainly by the tribal as well as the rural population, because these tribal communities as well as the rural population, they depend on the forest for their, mainly for their sustenance needs, fulfillment of their sustenance needs. And these wild plants and its parts are used by most of the rural Indian population, that is almost 85% Indian population, as traditional medicine, diet supplement, and for livelihood security as well as economic upliftment. The indigenous communities of both industrialized as well as developing countries fulfill their, depend on the wild plants for fulfilling their sustenance needs, and the main use of wild plants is 120 species per community. And that, as we know, the state of Jharkhand has a large tribal population, that is almost 26.3%,
सर जीमेल को एक बार ऊपर से नीचे की तरफ में खींचिए या फिर
a total of 32 tribes predominant in 17 districts out of the 24. Out of the 24 districts of the Jharkhand, in 17 districts, we used to get the predominance of tribal population. And as we all know, that indigenous communities reside in Jharkhand, in different parts of Jharkhand, has wide knowledge of wild plants, and they have developed unique knowledge of their utilization, mode of utilization. And the dependency on wild plants are quite high in Jharkhand, as forest is considered as one of the high value commodity across the state. Because not only in Jharkhand and in India also, we have the maximum population dependence over the agriculture, and we consider forest as our second largest land use pattern in our country. So in general, we know that tribal people used to live in close association with nature and maintain a harmony, maintain a connection between man and the environment. And it's a graph I have taken from one of my publications, that is Kumaran Chaitya 2020. So in this graph, I just want to show like what are the different plant parts used by the 4 different medicinal formulation. Here you can see like AP means the entire plants, whole plants, like Siwartia chiraita. This is one of the major immunobooster considered for the COVID-19 pandemic. And then edible leaves like Endocrinology album, then edible root like Curcuma longa, edible flower like Kerissa spinera, edible leaf and root like Kerissa opaca, then leaf and seeds like Spermococci articularis, and miscellaneous like Asparagus densiflora. So, as we all know, these Indian herbal and medicinal plants have a vast repository of bioactive components used in combating many diseases. And more than 30,000 plant species are edible, of which approximately 7,000 species have been used for nourishment as a diet supplement. wild edible plants, mainly the leafy vegetables, used to have high nutritional as well as medicinal values and are integral part of the daily diet of indigenous people of Jharkhand. Not only Jharkhand, even the people of West Bengal, people of Odisha, people of Northeast India, means where tribal dominance is there, in all those areas, we use these wild edible plants, mainly the leafy vegetable, as one of the diet supplements.
ये वाले लिंक से
So here just I am trying to show some of the photographs of the quite common medicinal plants of Jharkhand. Here this is Mesosphaerum suavulens. This is Asparagus sensiflorus, as I mentioned. This is Azadirachta indica. This is Heliotropium indicum. That is Erythema heterophylla. And this is the Asian marmelos. So now I'm going to talk about the indigenous ways to fight against COVID-19. As we all know, COVID-19 is a global health concern and everyone is focusing on preventive measures, at least until an effective cure is found. And according to WHO, the most effective ways to protect against coronavirus is by frequent cleaning of hand with alcohol-based sanitizer or washing with soap and water. So it is already, even in the advertisement also, it is announcing. And WHO also suggested that people should eat only well-cooked food and avoid spitting in public and avoid the huge gathering. And it is also important for people to seek medical care at the earliest if he or she become sick. So, as we all know, our government of India is also promoting the potential of herbal medicine as a part of the Go Local campaign. कुछ दिन पहले मोदी जी आए थे टीवी में and he was also suggested like we have to go local and we have to vocal for local. And in tune with the homemade remedies suggested Ayush Ministry to build immunity against the coronavirus. And Ayurveda experts have stressed the medicinal herb like Amla, that is Phyllanthus indica, Giloy, that is Tinospora cordifolia, Neem, that is Azadirachta indica, then Kutki, that is Phycorrhiza purua, and Tulsi, that is Oxymum sanctum, are helpful in strengthening the immunity to fight against this deadly virus and also preventing the infection of COVID-19. Again, like regular consumption of powerful Ayurvedic herbs boost the human body's immune system, which is considered as a foremost requirement to keep even the most feared communicable disease at bay. Medicinal plants can provide a cost-effective therapeutic option to cure these deadly viral diseases in humans with minimal toxicity. If someone has symptom of coronavirus, then consuming kara, that is a herbal tea or decoction of Tinospora cordifolia, Oxymum tenuiflora, with piperdigrum, that is black pepper, curcuma longa, turmeric, and gingiber officinale with ginger, that will boost immunity and kill all kind of viruses. The beneficial effect of Camelina sinensis, chai, even Ministry of Ayush has also suggested that if you try to be away from the coronavirus infection, then have tea and Terminilius sebula. This tea as well as extract from tea as well as this is hara. The combination is known as harad, have the potential to act as a therapeutic agent against COVID-19 infection. It is a new study done by the IIT Delhi. And this study also suggested that possibility of galotanine, that is hydrolyzable tannic acid extracted mainly from the plants to emerge as a potential therapeutic candidate against this SARS-CoV-2 in the coming future. Besides, there is a team from IIT Delhi's Kusuma School of Biological Sciences. They have screened almost 51 medicinal plants in 3-cell protease of the virus, and which is necessary for processing the viral polyproteins, and therefore emerged as an interesting premise for the development of drugs targeting the virus. The research found that targeting of this protein may be halt the replication of virus, means further infection can be halted by using these medicinal drugs. And individuals suffering from infectious and non-infectious diseases possess basically a lower immune system, and as WHO also confirmed, like the people who have lower immune system, they are at the higher risk to be affected by COVID-19. And the death of COVID-19 patient, it is again a report of WHO, the death of COVID-19 patient without pre-existing medical condition is quite low, that is only 0.9%. While the death of the patient with the pre-existing medical condition like cardiovascular diseases, diabetes, and chronic respiratory diseases is very high, range from 6.3% to 10.5%. So, as we all know, there is no medicine for COVID-19 pandemic till that. So, the only way to avoid this infection, we have to boost our immunity. You have poor immune system, then you are susceptible to COVID-19. If you have a better immunity, then you are less susceptible to COVID-19. So that's why the Ministry of Ayush also recommended certain medical decoction to fight against this COVID-19. And this Ministry of Ayush is like Ayurveda, Yoga, Naturopathy, Unani, Siddha, and Homeopathy. This Ministry of Ayush issued advisory where it recommended the use of herbal tea composed of tulsi, dalchini, that is, then kali-mirs, sunthi, and munakka for the self-care to develop immunity against this severe infection caused by this SARS-CoV-2 virus. And due to the complex composition of multiple phytoconstituents of all these medicinal plants, the mixture of this combination could module multiple proteins and would help to boost the immune system to contain the virus infection. And it also helps in increasing production of interferons, that is proteins, and antibody to generate an immune response against the virus and increase the rate of phagocytosis to destroy the microorganism. So further, this Ayush has also recommended the consumption of golden milk. Golden milk means haldi doodh, a half teaspoon of turmeric powder in 150 ml of hot milk once or twice a day. They also suggested for jaggery, use of jaggery, as well as fresh lemon juice to help to fight against the COVID-19 pandemic. They also suggested the use of sesame or coconut oil or ghee in both the nostrils in morning as well as in the evening. So here just I am trying to show some of the common immunity booster plants of Jharkhand. This is Tinospora cordifolia, that is giloy. This is Muraya koiniji, this is Karipatta. This is Sinopodium alba. and this is Amaranthus viridis. And when we talk about the common immunobooster plants of Jharkhand, as you all know, this is Siwartia syriata, jo locally known as Shiraita. It is a medicinal herb, and people use the above ground parts to make medicine, and it is used as a tonic to boost the immunity to fight against COVID-19. So nowadays, even in the local market, people are used to search for this Shiraita, and the demand for this Shiraita is increasing because of its immune boosting nature. It is bitter in taste, and it is used as a cure for impurities in blood, also in fever, constipation, upset stomach, of appetite, intestinal worm, skin diseases, and also in cancer. And in India, it is used for malaria when combined with the seeds of Cisalpinia bondeus. And the next common wild edible vegetable, leafy vegetable, that is Chenopodium album, locally known as Bathua Sag in Jharkhand. It is a unique vegetable known for its digestion aiding properties and used to body detox. And it is also used in abdominal pain, eye diseases, and in throat troubles. Next is about the giloy, that is Tinospora cordifolia. It is also quite common in our Jharkhand forest, and different plant parts including root, stem, as well as the whole plants used to prepare herbal medicines, and it is used for diabetes, high cholesterol, allergic rhinitis, that is hay fever, upset stomach, gout, lymphoma, and other cancers, as well as rheumatoid arthritis, hepatitis, peptic ulcer diseases, fever, gonorrhea, syphilis, and also to boost the immune system. Similarly, the other wild vegetable is bengsa. It is also common in our local markets in our Jharkhand, and scientific name is Centella asiatica. It is a medicinal herb, has a variety of benefits for the human body, and it is used as an appetizer and blood purifier, and also used in skin treatment in a wide spectrum of skin conditions. It is also used to repair nervous tissue due to the spinal injury, neuromuscular disorders, and also to increase general brain function as well as memory. So this Centella asiatica or bengsa, it is basically known for as a memory booster plant, and it is quite common in our local markets. It is also recommended for the treatment of various skin conditions such as leprosy, lupus, varicose ulcer, eczema, psoriasis, diarrhea, fever, amenorrhea, disease of the female genitourinary tract, and also for relieving anxiety, improving cognition, and also for the improvement of mental health. And during this COVID-19 pandemic, the demand for this is also increasing. And then another common leafy vegetable is Amaranthus viridis, that is locally known as Gandhari Sag. It is a leafy vegetable used as traditional medicine in treatment of fever, pain, asthma, dysentery, urinary disorder, liver disorder, eye disorder, and various other diseases. And this plant also possesses antimicrobial properties. So next is about the Phyllanthus indica, जो कि हम लोग locally आमला बोलते हैं. So these amla fruits are collected by the villagers and sold in the local market at the rupees of 5 to 6, and it is used in making sabankras, murabba, as well as achar, pickles. So that is after this value addition, we can sell this amla at the rupees 40 per kg. This amla protects cell against free radical damage and provides antioxidant protection, and it is used to treat skin disorder, respiratory infection, premature aging, and also useful in hemorrhage, diarrhea, dysentery, and has therapeutic value in treating even diabetes. So this is about the Andrographis paniculata. As I told you in the beginning, it is quite common in our Jharkhand forest, locally known as Kalmeg, king of beetles. And it is important plant species in Ayurvedic medicine, and it is prominent in 26 Ayurvedic formulations. It is an annual herb possessing immense therapeutic uses. Mostly used parts are roots, leaves, as well as aerial part of mature twigs. It is used to release body heat in fever. This species is effectively used as immunostimulant for asthma, gonorrhea, piles, dysentery, blood purification, influenza, gastric complaints, diarrhea, tonsillitis, pharyngitis, fever, hair loss, snake bite, common cold, diabetes, respiratory tract infection, as well as in jaundice. It also possesses anti-ulcerogenic and anti-typhoid, anti-snake venom, anti-platelet aggregation, anti-HIV, anti-malaria, anti-fertility, as well as anti-inflammatory properties. Next is about the Smilex zeylanica. It is also quite common in our Jharkhand forest. It is used as alternate source of chopachini, which is one among the top 20 highly traded medicinal plants in India. Because maximum chopachini used to be collected from the species Smilex chinensis, and Smilex zeylanica, it is used as an alternate source for this chopachini. It grows in hilly areas under moist evergreen and mixed deciduous forest from 200 to 1000 meter altitude, and also grows well in moist deciduous and semi-evergreen forests and in the plain areas. Its leaves and roots are used for medicinal purposes. The plant is considered as a remedy for syphilis, rheumatism, skin diseases, gout, venereal diseases, sore throat, dysentery, ulcer, gonorrhea, as well as pyorrhea. So stem are used as a toothbrush, which is considered useful against the toothache. Next is about the night jasmine. The scientific name is Nyctanthus arbotristis. It is commonly known as Harsingar. It is also quite common in the Jharkhand forest, and also it is used to be cultivated in most of the Indian garden as an ornamental plant. It is generally grows in tropical and subtropical region and distributed in some Himalayan regions. In India, it grows in the outer Himalayas and also found in the tract of Jammu and Kashmir, Nepal to east of Assam, Bengal, Tripura, extended through the central region up to the Godavari in the south. We can say like this Nyctanthus arbotristis is cosmopolitan in distribution. In case of India, from south to north, from Himalaya, Jammu and Kashmir, western Himalaya to the eastern Himalaya, it is extended throughout the country. So it is one of the well-known medicinal plant, and different plant parts are used in Indian system of medicine for various pharmacological action like anti-leishmaniasis, antiviral, antifungal, antipyretic, anti histaminic, anti-malarial, antioxidant, anti-inflammatory, as well as anthelmintic, because its leaves as well as flower are quite bitter in taste. Next is about the polar henna pubescens. It is one of the IUCN red listed species common in our Jharkhand forest. It is a least concern species and it is an Indian medicinal plant that has anti-dysenteric, anti-diarrheal and anti-amoebic properties. Actually, initially the name of this plant was Polarhina anti-dysenterica due to its effectiveness for curing dysentery. But the latest ISBN has modified its name as Polarhina pubescens, and in Ayurveda it is used for the treatment of amoebic dysentery, diarrhea, as well as irritable bowel syndrome, bleeding piles, as well as liver disorder. The root boiled in milk is applied against snake bite and is used in the treatment of venereal diseases. A hot decoction of the stem bark is used as a gargle to treat toothache. The seed or bark are sometimes adulterated with Riccia tinctoria due to their antibacterial activity. Then again, next is about Pterocarpus marsupium. It is again an IUCN red listed tree species. It is a near-threatened tree. In case of Jharkhand, it is commonly distributed in Ranchi and its nearby districts. And it is extensively used for the treatment of diabetes, obesity, diarrhea, vitiligo, and psoriasis. The bark is also used for bleeding as well as in toothaches. The leaves are often applied externally as a remedy for skin diseases, and the most common used in modern herbal medicine include to help support the body's natural ability to manage and regulate blood sugar level. So that's why it is considered one of the major components of diabetic treatment. Another IUCN red listed species common in our Jharkhand forest is Gloriosa superba. It is also a least concern species commonly found in Hazaribagh, Gumla and Simdega. It is in great demand for medicinal use, so it is cultivated on farm in India. But most plant material sold into the pharmaceutical trade comes from the wild population. It is also widely distributed from south to up to east northeast, that is in Assam. It is quite common and it is used traditionally for the treatment of colic, chronic ulcer, hemorrhoids as well as for cancer, and it is employed as a tonic and purgative. So this is some of the important plant species I have mentioned that is quite common in our natural forest. So our natural forest, it's facing major challenges due to firewood collection, timber felling, mining, herbivory including grazing and insect herbivory, fire, anti-species collection as well as invasive species. So these are some of the photographs taken during our field visit to the forest of Ranchi, and this is again timber cutting. This is mining and grazing. Here rock mining, soil mining, along with the various metal and mineral mining, it is quite common. And besides, various animal grazers are also common in our natural forest. Besides that one, the forest of Jharkhand, it had been invaded by several invasive weeds like Ageratum conijoides, then Promolina odorata, Hyptis suaviolens, Imperata cylindrica, Mimosa pudica, Parthenium hysterophorus, Lantana camara. So this is lantana, this is hyptis, and this is parthenia. These are then this is mimosa pudica, and in the background that is Cromolina odorata. And it's a picture taken in the Scilly forest range. You can see inside the forest we are getting a heavy patch of invasive species like mimosa, hyptis, as well as Cromolina odorata. So besides that, in our Jharkhand forest, we have various common insect pests which are causing great damage in the growth of seedling as well as various tropical plant species. So in conclusion, I just want to say like intake of the multiple compound in the form of herbal tea as suggested by Ayush could be more beneficial rather than a single molecule for boosting the immunity as prophylactic against COVID-19. And it would be helpful for those who are suffering from diabetes as well as in hypertension, because 4-hydroxychloroquine treatment cannot be proceed mainly for diabetic as well as hypertension, that is high blood pressure patient. So as you all know, till that there is no medicine for this COVID-19 pandemic, and whatever treatment is growing out throughout the globe, mainly on the basis of 4-hydroxychloroquine, it is an anti-malarial drug. So a variety of medicinal plants are grown in the forest of Jharkhand, but due to the unscientific collection, poor knowledge, and unavailability of proper management practices are the reason behind the minimum economic benefits of the local people. But as we all know, that scope is unlimited. The diversity of wild edible plants in Jharkhand is depleting due to the over-exploitation, unsustainable harvesting for food, medicine, as well as due to various biotic interferences, including grazing, herbivory, as well as anthropogenic fire. Because in Jharkhand, we use the flower of mahua, that is Madhuka longifolia, for the preparation of traditional alcoholic beverages. So, for the collection of the flower of this mahua during summer months, March to May, that is generally considered like what is called pre-monsoon season here in Jharkhand, and most of the plants used to start regenerating. And at that time, for the collection of mahua flower, this local indigenous people used to add fire in the ground covers so that they can easily collect the flower of the mahua for the traditional alcoholic beverage preparation. So therefore, there is an urgent need to conserve these valuable wild edible plants and use it in a sustainable manner to ensure the future demand. Because as we all know, right now we have no medicine for COVID-19 pandemic, and that's why people are searching for various options among the traditional herbal medicines. So it has huge demand, and who knows tomorrow which kind of pandemic is going to be rise. Because as you all know, this humankind is alarmed by various deadly diseases from time to time, like AIDS, hepatitis B, then what is called SARS, SARS, as well as like this bird flu, swine flu, and presently this COVID-19. So at last, I just want to conclude with the WHO's One Health initiative that seeks to promote the optimum health for people, animals, and the environment, and it advocates managing the issue of human health in relation to the environment as well as biodiversity. And this WHO's One Health initiative has 3 major objectives. Number 1 is combating zoonotics like this COVID-19 pandemic, Ebola, as well as ensuring food safety and fighting antibiotic resistance. So this is all about my presentation. Thank you so much, and thank you to Dr. Ima for giving me invitation to give a deliberation among you people. So if you have any query, you are free to ask.
Excuse me, ma'am. Thank you very much for this enriching session. It was indeed a very good one. Here is one question, ma'am. There are the medicinal plants you are talking about. They are cure for so many diseases. Still, people are using synthetic medicines. Why is so? One question is here in the chat box.
Okay, actually what happened in case of ethnomedicine, because our topic is like ethnomedicine. So when you go through the literature of the ethnomedicine, then you will get like, means people used to say like you have to take like for immunity boosting against the COVID-19. They are telling, Ministry of Ayush is also telling like preparation of herbal tea with giloy, with curcuma longa, what is called turmeric, then kalimirch, adrak, tulsi. It has mentioned like that, but there is nowhere it is mentioned how much of tulsi leaf is required, how much giloy leaf is required or giloy root is required. Nowhere it has been mentioned. So that is the reason actually when we used to take these ethnomedicines or traditional medicine, herbal medicines, due to the proper value addition, due to the lack of proper value addition, we are not aware what is the actual amount is necessary for recovering or for boosting the immunity, for recovering from any diseases or for boosting the immunity. That is the main lacuna in the traditional medicines. And people used to say like the 2 leaves of harsinger, you dip in water and take the decoction. 2 leaves, that is smaller leaf, medium size leaf or long, that was larger leaf. So there is no proper what is called composition is there for the traditional medicine. That's why although they have various medicinal perspective, due to the lack of proper what is called combination, proper composition, it is sometimes used to take so many time to recover from the diseases. So that is the reason why people used to believe on more on allopathic medicine as compared to traditional herbal medicines.
Thank you very much, ma'am. Thank you. Really, it was an enriching session. It was a wonderful one. Thank you. Once again.
Thank you so much, Dr. Ima.
Yes, over to Nikki.
Thank you, ma'am. I request Rituvi Prasad to give thanking speech to Saikia ma'am.
Good afternoon to all. On behalf of entire fraternity of Nirmala College, I extend my hearty thanks to you, ma'am, for making this excellent presentation and making this workshop meaningful for us. I hope all the students would definitely have been benefited. And thank you, ma'am, once again for sparing your valuable time. Thank you.
Thank you so much, Rutvi, Nikki, Dr. Ima, and entire organizing committee members, fraternity of Department of Zoology as well as Department of Botany. Just I want to share here, I am, although teaching here the environmental science, I am basically from botany background, and whatever profile you have today talked about me, that is actually 2016 or 17 old profile. So thank you so much. Thank you so much for everything, for inviting me, for delivering the talk on the immunobooster plants of COVID-19. Thank you so much.
Yes, ma'am. This will be a long-lasting connection. Now we will be up to you for anything for ethnomedicine. It was really a nice one. Thank you very much.
Thank you so much. Thank you so much.
Thank you, ma'am. So we'll continue with the first session, which was Sericulture.
And I welcome Dr. Jayaprakash Pandey, CTRTI Senior Scientist. Sir, kindly start the session.
Yes, madam. Am I audible now?
Yes, sir, you're audible.
Yeah. First of all, I am very sorry because morning due to some technical difficulty, I could not able to present my presentation, and now possibly this got rectified. Now I am going to tell something about my topic and what are the main theme of this seminar is the exploring the science behind the sericulture. This will be the main focus of my presentation today. As we know that sericulture is one of the industry which is spread over various part of India, and there are many variety of the sericulture insects are available in nature. And if you see the variety wise, we have mulberry silkworm, that is Bombyx mori, tussar silkworm, that is Entheria malata, muga silkworm, Entheria asamensis, and iris silkworm. Now, if you see the map of the India, you can find the various variety in various locations. But place Jharkhand is the place where maximum production will be there from the tussar silkworm, that is known as Entheria malata. Total production of the country is around 3,268 metric ton. Out of that, around 2,600 metric ton is produced in India. That's why this Jharkhand is also known as Tussar capital. And not only that, if you see the map of the various what you call the in product map country, you can find various dresses and various specific print from the various part of the world, but various part of the India. And the design is specific to particular pockets, and people are earning good amount of money out of doing this what you call sericulture aspects. And India is the country where all four varieties like mulberry silk, tussar silk, muga silk, and iris silk rearing is conducted, because we have diversity in our country. We have various types of climatic conditions. That's why we are able to rear all the varieties. And one very important point I would like to mention. After China, India is the second most producer of the country. But if you see the production of the China, that is around 1 lakh metric ton. But production of the total silk production in our country is around 30 to 34,000 metric ton. So still there is huge gap. That's why I am very happy that now students are showing their interest in tussar sericulture as well as sericulture, which will be very much beneficial in future, because if many people will join with tussar sericulture or sericulture as a whole, then a lot of benefits will come in society, and one day will come our actual aim will be fulfilled, because we will be having global silk leader in the world. So now I'll focus on my topic, that is science behind tussar sericulture, possibility of education and employment in the country. And as we know that the rearing of tussar silkworm is conducted on three main food plants, that is Terminalia arjuna, Terminalia tomentosa, and Surya robusta. And there are many secondary food plants are also there, but because of availability of the these plants, Arjuna, Asan, and Sal, we are mainly focusing on these three primary food plants. As we know that, very interesting fact about the tussar sericulture, to produce 1 gram of silk, insects consumes around 280 to 320 grams of the tussar, their food leaves from their food plants. Like to convert 1 gram silk protein, insect has to feed on Terminalia tomentosa and Terminalia arjuna, and they have to consume around 320 grams of the leaves. Now you can see that what is the conversion ratio. And second very important points I'd like to tell you, because one silkworm's eggs will be around 8 to 10 mg, and that silkworm after voracious feeding on during the first instar to fifth instar, they will get converted from the 8 mg, around 8 to 10 mg to 40 grams. You can see how much conversion is there, 40 grams. And from 40 grams larvae, we will be getting only 1 to 1.5 grams the silk. That's why this is the beauty of this industry, because this industry provides the 90% rural employment. That is very, very important. Now, there is a big challenge for our country to provide the rural employment. Even not only rural, this core focus is the tribes population, because whoever resides nearby the jungles and where Arjun Asan and Sal plantation is available, they are getting benefited. And one progressive farmers who will be doing Tassa silk for rearing, grainage and other aspects, they can get even 50 to 60,000 per year out of doing other activity in their family. Like apart from doing agriculture aspects and their daily living, they can earn around 50,000 from the Tassa Sericulture. And second very important point is post-plant availability. There is huge quantity. Still, we are producing around 3,400 metric ton total silk in country, but we are able to utilize only 5 to 6% of the Tassar flora. You know, there is still lot of scope to get production and productivity much better than what we are getting now. Now, if you see talk about only Tassar Sericulture, around 3.5 lakhs people are associated in country. And more importantly, these people are initially they are very poor. Now they are reforming their life to Tassar culture. That is a very, very important aspect. And third, if we talk about any industry, it means industrialization and industry means that is anti-environmental aspect. But here that whenever Tassar culture we promote, we are going to do plantation, means that is eco-friendly industry, and that is very important for the climatic aspects also. And rural women employment, tribal employment, and most important point is the low investment, high return. That is very, very important point of the sericulture industry. Here, person who is interested to initiate their business, they can initiate only with the 4 to 5,000. That money is very less, and within 40 days they can earn around 40 to 45,000 rupees, and this is very important. And as you know that throughout the year, the rearing will not be conducted. There will be 2 crop systems in the country. That is, one will be initiated in month of July, and second crop will be initiated in month of September. Now, this is a second crop is going to be initiated, and from one cocoons to produce one cocoon, person used to get around 3 to 4 rupees. And DFLs which they get it, one DFL is equal to 200 eggs. From out of 200 eggs, they can produce even 100 cocoons. And 100 cocoons means 100 cocoons they can get it 300. Means by spending 12 rupees, they can earn 300 rupees. This is the beauty of this industry. Means lot of scale up will be there. And one very important aspect regarding this industry is that money flow in this industry comes from rich to poor. That is very important. Majority of industry, money will go from poor to rich people. Here, because many people are involved in Tassar Sericulture industry, still this is labor-oriented industry. So that it is providing lot of employment in rural people. That is very important people and a very important aspect. And they are getting money from rich people to poor people. And if you see the whole scenario of this industry, like success, if you talk about the science behind this Tassar Sericulture, the main success depends on feed, means what quality feed we are providing. That we can see here that terminal tomatoes are plants, and you can even have terminal Arjuna also, Surya Robusta also. Feed means you need to have the good feed quality, and if feed quality is good, then you can produce better seed, and if seed is better, then you can produce better breeds. And breed is very good. Then through the management aspects like disinfections, using the required quantity of fertilizers in the field, you can get better crops. Now, important point is that what are the components of the leaves? Why Terminal Arjuna is not Anthera mariata is not feeding on other food plants? They are feeding mostly on Arjuna and Sal, because they are having some interaction, like insect plant interaction is there. That's why they are feeding on the only on Termina arjuna, Termina tomentosus, robusta, and some of the secondary food plants. Now, one very important aspect I would like to tell you, like whenever we initiate the rearing, we say the total silk scenario soil to silk. Soil to silk means we have to initiate our sericulture starting from the soil enrichment. Soil enrichment, there are two ways. You can use chemical fertilizer, but nowadays we are not advocating the chemical fertilizer. To minimize the use of chemical fertilizer, we are now disseminating the technology of vermicompost, farm waste technology, and by using the other green manuring like sunham dhecha, so that we can enrich the soil. Once you enrich the soil, the same quantity of nutrients will go to insect, and those insects, if they will produce the what you call cocoons, those cocoons will be good quality, and they can earn even much better than what present system is there. Now, rule of our institution, as we can see that the production in our country, it was 428 metric ton. Through the technical intervention, dissemination of technology, and workshop like you are conducting, we have disseminated our information to the rural people. Now we can see that the production of Tasar sericulture has increased, starting from 2007, starting from 2007 to 8 to 2019-20. You can see the 3,515 metric ton. This is very important aspect. Now, what are the various steps involved? Like first of all, you have to do plantation, and after plantation, you can get seed from our seed unit, and rearing will be initiated. After initiation of the rearing, this larva will get converted from first instar to fifth instar. Afterward, cocoon will be produced in outdoor condition, and cocoon will be collected in indoor. And afterwards, cocoon will be dried, reading will be conducted, spinning will be conducted, and degumming. Prior to coloring the what you call fabrics, we need to perform the degumming also. Why degumming we say that? Because silk is made up with two major proteins. One is known as fibrin, and second is sericin. Fibrin will be around 60 to 70%, and sericin will be around 15 to 20%. So sericin is known as a gummy protein, that is having lot of gums, and so many other minerals will be also available with the cocoons. That's why prior to what you call coloring the fabrics, we need to perform the degumming of the cloth. That is a very important steps during the preparation of the fabric. So ultimate through these steps, around 6 to 10 steps, ultimately silk fabric will be produced. Now, if you see the various steps, what are the value additions we are doing? You see, one lady is wearing the silk sari, that is Tasar silk sari, and you can see the various steps like eggs after the feeding, first to second star, third star, fourth star. Afterwards, cocoon production, and they will sell the cocoons in market. Afterwards, that spinning will be conducted, and weaving will be conducted, and ultimately fabric will be prepared. Lot of labor-oriented work. Now, this industry is having labor orientation because now under this COVID-19 situation, many people got they are not in job now, because some of the people have lost the job, and many people migrated from their job places. So now giving job to those people are very big issue. Now in Tasar industry, what we can say that we can provide job through the Tasar sericulture, and climatic condition of the Jharkhand is very effective for the doing the rearing of the Tasar silk worm, and that's why we are focusing on Tasar sericulture. Now, what are the steps? One more step is that, like in seed production. Seed is, as I mentioned, that seed is one of the very important aspects in Tasar sericulture industry. In this, emergence of moth will be there. We can see the many male and female moths here. After that, male and female moths will be coupled. They will couple, and after that, this egg laying will be there, and this seed will be utilized. Now, one important step during this, what we used to do, one disease which caused the loss to this industry around 20 to 30%. To minimize that disease, what we have to do after egg laying, those mother moths will be examined through microscope, and if those moths are suffering with some disease, then what we can do it, we can discard those lards for we are not going to utilize for the seed production. Only moth which is pepprin free, that will continue for the next generation seed production. Now if you see the diversity in the silk and diversity in their cocoons, Intheria malata is only one genus species, but they are producing various morphotypes of the cocoons, and that's why many ecoraces they are acclimatized to various pockets of the country. That's why that is known as the various ecoraces of the Entheria malata. You can see the very small cocoons here, two very big cocoons. All are Entheria malata, but their morphology, their cocoon characters are different. And Jharkhand is having a very rich flora, and out of 44 ecoraces, even 10 are present in Jharkhand. And that is very important. And one of the ruling variety, that is Dava ecoraces, that is origin place is the Saranda forest. And ultimately, there is a lot of evidence we can say that the origin of Tasar silkworm Entheria malata happens in the Jharkhand only. There is a lot of evidence regarding this. Now you see the scenario where ethnic people they reside, and they used to perform the conjuration of the plants, conjuration of the silkworm also in natural condition. And what we have been, what we did it, we have released our population, huge number of population in the Palma forest of the Jharkhand. And what we find that they are able to multiply their numbers in those forests. Nowadays, we are getting huge collection from that. But while doing the collection, we divide those food plants, those areas in 3 zones. One is core zone, and second is buffer zone, and third is periphery zone. We used to collect the cocoons from the periphery zones. We are not focusing on the core zone, because once you collect the cocoons from the core zones, there will be no population available there, and there will be decrease in production. That's why we we collect the cocoons from their core zone only, their peripheral zone only. Now, what are the reasons? Like one silkworm will produce around 200 eggs, but still we are able to harvest 50 to 70 cocoons per day. Means out of 200, we are able to produce only 50 to 100 cocoons. Where hundreds are going? Where other hundreds are going? When we have documented that, we find that there are 4 major diseases: viruses, pepprin, and muscardine, and bacterosis. These 4 diseases are affecting the Tasar silkworm rearing, and due to these diseases, around 40 to 50% loss occurs. And apart from that, several vagaries of nature, high temperature, high humidity, heavy rainfall, because the silkworm, Tasar silkworm rearing will be conducted outdoor only, and there will be a lot of loss occurs. So what we are doing, we have developed the several technology also. We have developed various products also, like Jivan Sudha, LSM, to minimize the impact of these disease. You can see the symptoms. You can see the this is nosema infected, that is microsporidial disease, and viruses, and muscardine disease, and bacteria. These are the symptoms. We can see the pepper-like spots on the silkworms, on Tasar silkworm. And you can see the this Tasar silkworm larvae infected with the virus, that is a cytoplasmic polyhydrosis virus, and this is the symptoms. What we have to do it, whenever silkworm is infected with this disease, we have to collect the worm from field, and proper disposal should be there, so that this will not affect other insects also. Likewise, for other disease also, bacteriosis, muscardine, and nosema related disease also, we are doing same kind of scenario for that. As we know that Tasar silkworm rearing is conducted in outdoor condition. So what we are doing, as we say that we give the slogan, prevention is better than cure. So what we have to perform, we have to perform pre-rearing disinfection. Wherever we conduct the rearing, prior to rearing, we conduct the disinfection, and after conducting the rearing also, we conduct the disinfection by using the flame gunning, by using the bleaching powder and chuna, and so that disease concentration will be low. Now in this slide you can clearly see that spores, that is nozomi spores, and these are the various spots as I have already explained that, and these micros these spores can be detected whenever you are performing the microscopic examination. So what we are doing, if this kind, you can see these spores like rice grains, and whenever these kinds of spore will be visible in our microscope, even one spore will be there, those lot will be discarded. Only pebrine free lot will be utilized for the industry. Now there are many symptoms. Like one I tell that what you call, there will be spots on their skin. Second, irregular growth, and many other symptoms will be there, so that we can identify that this insect is suffering with the pebrine disease. Now, likewise, viruses, as we can see the symptoms, and after this symptom, virus is one of the viruses is one of the very lethal disease for the silkworm. Whenever this viruses come, when humidity will be very high and temperature will be also very high, this viruses will occur, and this is the what you call 20 to 30% loss in normal condition, but sometimes even 80 to 90% loss occur due to the virus. That's why we need to be very careful with this cytoplasmic polyhydrosis viruses while doing the rearing. Now, bacteria-related aspects, I already explained the symptoms, and muscardine disease also. This is also fungal disease, and this already I have explained. Now, what we have to need, what we need to do it. Now, number one point is that proper disinfection of the field. Second, pre-rearing disinfection, post-rearing disinfection, and persons whoever are doing the rearing, they should get training from the orange school, so that they can perform the rearing perfectly and with all the package and practices. Now, what are the practices we are doing? Like we are providing the quality seed to farmers and disinfections, and we need to maintain the hygiene condition in the field. As I mentioned that bleaching powder and tuna, pre-rearing and post-rearing disinfection, and during rearing also, we cannot utilize any pesticide or any disinfectant on the leaf, because if silkworm will feed, then lot of loss will occur. That's why we need to be very careful. If any insect is suffering with disease, immediately we have to take it from field, and so that disease percentage can be minimized. And you can see in these photographs, person who is using bleaching powder solution, 2% bleaching powder solution, and spraying in the field prior to performing the rearing. And second part, they used to perform the flame gunning. This is also very important aspect. Flame gunning to various viruses, various pebrine disease will be there, and those disease contamination will be less if you perform the flame gunning in month of April. So this disinfection, you can see the here application of the tuna prior to rearing, and the man is doing. And second very important aspect related to this, transfer of the larvae in the required quantity. Like if you not perform the rearing as per the plan, as per the real quantity, you may have problem. One plant, if you provide per plant around 100 larvae, then that will be sufficient for that. Otherwise, what happens if you put more plants, there will be lot of chances for the disease occurrence. Now, second part is that silkworm litters. Silkworm litters are also part of contamination. Whenever this rearing you are conducting, day by day cleaning and their disinfection by using bleaching and tuna can be conducted by sprinkling of the bleaching powder and tuna. And last, very important point is that while performing rearing, in between also we perform the microscopic observation, so that if any disease is occurring in Tasar silkworm, you can minimize the dose of those disease, and you can even perform the rearing in better way. So while doing rearing, if disease is there, then we will be having two options. One is there, if silkworm cocoons are infected with the pebrine disease, then those cocoons will be stapled and utilized for the fabric preparation. And if seed quality is, cocoon quality is very good, then we can go for the seed production. Only pebrine and virus free lots should be utilized for the seed production, so that we can minimize the minimize the pathogen loads. Now, these are the various steps involved during the sericultures and various mixtures, how to prepare, how to perform the like flame gunning and cleaning, disinfections, and other aspects. Now, if you see the total scenario of the sericulture industry, like involvement of the various age groups of the people in different activity of the like nursery, rearing, seed, reeling, and weaving. Now, this is the Tasas culture is a culture where you can provide the employment to each and every groups peoples. Like for young people, they can prepare the nursery, and from nursery technique they can get even 50,000 rupees rearing for age peoples like who's having 50 to 60 years, they can perform the rearing. Seed production, once again, this is focused on the young people. Reeling, even this sector is dominated by the women. Mostly they perform the reeling, and they perform the weaving also. These are the sectors which is mostly dominated by them. So what I mean to say that this Tasa sericulture has been employment to the poor people, not only to poor peoples, even person who is residing nearby the jungles, they are not having job, they can join this Tasa culture for their self-employment. Now, if you see this sapling raising, we are having technology for the sapling raising of the Arjuna plant, Asan plant, and one more plant, that is Jerul plants. By using this sapling, they can get even 50 to 60,000 by preparation of the nursery. Now, second point is that if someone, like once you initiate the rearing and plantation, then it takes around 3 to 4 years to grow. Meanwhile, what we can do it through intercropping also, like turmeric, ginger, horse gram, and many others you can utilize for the additional income for the farmers. And they can do even rearing. Abhi Prime Minister ji ke slogan tha, har med pe ped. So likewise, even while doing rearing also, you can get these products also, like by using turmeric and ginger also. Through intercropping, there is a lot of scope you can get it. Now, if you see the opportunities and potential, if person who involves with the seed production of the Tasar, they can get 18 to 25,000 in 15 to 25 days. There is a lot of scope. And two crop rearing, a person who is doing two crop rearing and producing the cocoons, they can get 50 to 70,000. And third important point is that is reeling. Reeling is very important aspect, and this is a mainly focused on women, because tribal women and many peoples nearby the jungles and other places, they are not having employment. And for them, this is very important aspect. We are having 5-6 variety of the machines available with us. Like if a person is having very less income, they can initiate even reeling with the matka. If they can go the various aspects of the reeling, and through weaving also, through even through local trades also, they can earn the money. Now these are the scenario I am telling the related to SHG groups. They are getting a good amount of money out of doing that. You can imagine here the one lady who is doing a microscopic observation to observe the pebrine disease. She is having training and she can do it. And with the help of this training, she is able to produce the DFLs, and she is able to produce DFL, and they are getting the money also, like 50 to 80,000 they are earning from this. Now, what they can do it, now if any entrepreneur, they want to initiate that, they can farm the SFG, and even they can get aid from the state government also, they can get aid from the NABARD, and they can initiate it. Now, what are the various programs? Various programs are also available, like Center Sector, IVLP, MKSP, MNREGA, to help them. And with the help of these programs, even they get machine also with the 90% subsidy. You know, if some machine costs 20,000, they need to pay very less amount, and they can get it. And if you see that person who involved in Tasa sericulture, they got lot of employment in there, and they have employed other peoples also. And you can see the scenario of seed production unit in the village area, and this is photographs taken from the Karsma region. and the person happy farmers who is going to sell the cocoons in the market. Now you see the cocoon harvesting and cocoon staplings and various steps involved the post cocoon sector. Like they have to involve, you start from the harvesting of the cocoon, stapling of the cocoons, storage and sorting of the cocoons, cocoon sorting and weaving and other activities. Now you can see that not only this, we are utilizing the solar operated machines. Even village area where electricity supply is not there, regular supply is not there, they can have electricity. We have developed a machine like various machines. They can utilize those machines, and the cost of the machine is also not high, very high. And apart from that, we are giving subsidy from our side. You can see the various machines like Kamdhenu, MRTM, MTRM for the preparation of yarns. These machines are any person who is going to initiate common facility center for the silk, they can use these machines. Now you can see the involvement of the rural peoples through Tasar cultures. Now a lot of peoples, even you can see that matka reeling also is very popular in Jharkhand. They can get even 200 to 300 out of doing this reeling, and if they enhance their skills by getting training from our institution, even they can get better. So what my main message to the various students, like getting job for the all people is very difficult in nowadays. What you can do, you can create the job through sericulture, you can create job through sericulture, and this is a industry where you can initiate your business with a very less amount only, 4 to 5,000, and even you are having lot of scope to make the various diversified products using the Tasar also. So and another area is byproducts. I am not going to go in details. Like byproduct is also very important aspect for the Tasar sericulture industry, and where by utilizing the byproducts, various useful products can be created, and they can even still enhance their income. And many proven models are also there. Beautiful beads prepared from the silk pupa, and out of that, from the Arjuna, we can produce the Arjuna powder, Arjuna Chonpras, even Arjuna with honey, and there are many business avenues. Any students, any faculty members who are interested to develop their entrepreneurship, they want to get training, they are most welcome, and they can visit our institution to get this training. And ultimately, what I want to conclude that Tasar culture can be done with the agriculture also, and there is huge opportunity for the self-employment, and the additional benefits through the intercropping can be done while doing Tasar culture also. And we have developed many technology, we have patents also, and there is huge scopes and availability, and we are having many scopes, many helps, and we are ready to provide the training as well as the facility from our institutions. We are having good collaborations also from the various organizations as well as from your organization also. I am very happy that Nirmala College is coming forward for their this collaborative aspects also. We are going to initiate the various projects with the Nirmala College, and I am happy that day will come. The many students will join Tasar sericulture, and ultimate production will be production of the Tasar sericulture will be very high, and many people will join, and ultimately India will be the global leader of the Tasar sericulture. This is my word. Thank you very much.
Thank you, sir.
Thank you, sir.
Yeah,
Sir, we have
I'll welcome any questions related to this my topic.
Students, you are unmuted. Kindly ask the questions.
Excuse me, sir.
Yes.
We have a question from Ankit Srivastava,
Yes.
who is known as the Manchester of India.
Manchester of the India.
Yes, sir.
Yeah, for the silk production. Karnataka
Yes,
is the
sir.
state. Karnataka
Okay,
is the state.
Karnataka state.
Karnataka is the state.
Okay, sir.
And the
Sir,
maximum production of sericulture that comes from the Karnataka. But if you talk about the Tasar sericulture, Jharkhand is the Tasar capital. Out of 3,600 metric ton, around 2,600 metric ton will be produced by this state only, Jharkhand.
okay, thank you, sir. Excuse me, sir. There is another question
Yeah.
from Harshita Kumari.
Yes.
Silk is biodegradable or non-biodegradable?
Yeah, biodegradable.
Okay, thank you, sir.
This is protein. This is made up from two proteins, that is fibrin and sericin, that is biodegradable. And one very important point is that silk is silk gives, especially Tassar silk, gives the cooler feeling in summer condition. And this will be warmer in the winter season. That is very specific character of the Tassar silk.
Okay, sir. Sir, we have another question from Neha Kumari.
Yeah.
What can we do for increasing production in core zone?
Yeah, what we are doing, like in traditional way, we have developed around 44 technology to disseminate in the field level. What we are doing the conservation of the silkworm. Like whenever we are collecting the cocoons from the core zone, then what happens that if core zone will be vacant, then ultimate production of the Tassar silk will decrease. So what we are doing every year, we are able to give DFLs, digital links, and we are relating to those particular fields where their core zones are there. And apart from that, we are doing conservation also aspects also. In those core zones, people will not be allowed to collect the cocoons from the core zone, so that their minimum population will be maintained in those areas. And what we are planning to further increase the production, because we are disseminating this technology to new field also, and we are performing the plantation also. Through economic plantation also, we can do the systematic rearing and we can enhance the production.
Okay, sir. Thank you, sir.
Any
question, sir? Hello, sir. There is one more question.
Yes, ma'am.
Divya Kumari is asking, what are the proteins that are removed during degumming?
Yeah, that is a sericin protein, madam. Sericin. Actually, silk is made up with the two proteins. One is fibrin and second is sericin. Fibrin concentration is around 60 to 70%, and this sericin concentration is around 15 to 20%. During the degumming process, this sericin will be removed, because fibrin is nothing but cloth. That is the main content. And sericin will be there along with the this silk, that is fibrin, to provide the strength. And still, whenever we prepare the fibrin of the fabric of the Tassar silk, during that time also around 3 to 4% sericin will remain there to provide the strength to the silk.
Thank you, sir. There is another question again. What is the eligibility to join sericulture?
Yeah,
it is asked by Sneha Kashyap.
Yes, a very important question, and I welcome many students if they are interested. Our institute, they conduct a postgraduate diploma in sericulture. Okay, after graduation, after biological science person who is having BZC group, they can join this organization, and there will be 18-month course here in our organization. After that, they can get a job in their respective organization and respective states also. And there will be two kind of vacancy will be filled here. One is the postgraduate diploma sericulture is sponsored by the states, and even students who are not sponsored, they can also join, but they have to bear their expenses. And whatever course we are conducting, we are getting sponsor candidates from the various part of the country, like Nagaland, Manipur, and various states, and they are getting training. And after this training, they can apply Central Silk Board, like technical assistants and other position in Central Silk Board. Apart from that, if they are MSc, they can apply for the scientist post also.
Thank you, sir. Gunjan Tiwari is asking the difference between machine spinning and man-made spinning.
Yeah, machine spinning. Okay. Whenever this spinning will be conducted by using machine, like MRTM and various machines I have already showed to you, the quality of the yarns will be very high. And those yarns will be sold out in market around, they can get even 5,000 rupees per kg. But yarn which will be prepared manually, they can get only 2,500. That is difference.
Thank you, sir. And then Sneha
I'm happy,
Kashyap.
madam. Many questions are coming from the student side, and if any query, I'm available. Please.
Yes, one more is here, sir. Ashwarya is asking, what is reeling in sericulture? Please, just answer.
I'll explain. Actually, whenever we produce the cocoons, okay, cocoons, after production of the cocoons, we need to take the threads from that. Okay. And in one cocoon, around 1,000 meters of the silk threads will be there, continuous. continuous 1,000 meters. Okay, around 1 kilometer. Even some of the silicone races can produce even 3 kilometers also. Prior to preparation of the yarn, we need to reel those. We have to take out those silk from the cocoons. That process is known as reeling. Yes, madam?
Thank you, sir. Sir, means my students, they are very much interested in knowing how to join sericulture, what are the marks required for that. I think if you will avail us some type of document, some prospectus or something like that, I will share it with my students. Yes.
What I'll do it, madam, we have one training program modules from our organization. Okay.
Okay, sir.
In that, there are various types of training programs available in our organization, and we can set the details. What are the course they can join? What are the course fee in that? And what are the terms condition in that? And what are the benefits to them? Okay, and we can send the, all the details to them. And anytime students are most welcome from your organization, because your organization is having a collaboration with CTNDI Rachi, and we'll be happy to even have exposure visit, even meetings also. And many times your fashion designing department, many students they have visited, and even some of the students, group of students, because this COVID-19 situation is there, even up to 20 groups they can visit. And prior to that, you can send one information to our organization, and you are most welcome, and we will be happy to show them all the activity. Even our museum also, the Silk Museum, which is unique in country.
Thank you very much, sir. Over to Nikki.
Yeah.
Thank you, sir. Now I request Neha Kashyap to give vote of thanks.
Good afternoon, one and all present here. I express my sincere thanks to our Honorable Resource Person of the Day, Dr. Jayaprakash Pandey, on behalf of Nirmala College, Ranchi. He gave a wonderful speech. See, sericulture is a part of our CBCS syllabus as a skill enhancement course. Of course, this knowledge would be beneficial to us in near future for self-employment purposes too. We appreciate your effort and presence. It was a wonderful lecture with loads of knowledge and information. I would also like to extend my gratitude to all those who sincerely committed to this workshop to make it a success. Thank you, everyone.
I am once again very thankful to Madam, Sister Jyoti, Principal Madam, and Madam has showed a lot of interest in Dasar Sericulture, and she visited our institute also. Madam, I request you, if you find time once you visit, because this active training will be going on in the month of September, and end of September, even first week of October, we can show you because last time when you have visited, you could not observe the whole value chain addition. Now it's active training period, madam. When you can come with the faculty members as well as student, and we'll have all the soil to sell activity, and we'll show to you, madam. So I'm very thankful to you for organizing this kind of workshop, and which is many students they have joined, and our ultimate aim is getting fulfilled, because we need to disseminate the technology. We feel that for the as per our the service also, so that this technology will go to many rural people, poor peoples as well as students, then much more benefits will come and people will get benefited.
Thank you, sir. Thank you very much, sir.
Yeah.
Thank you, sir. Once again, with immense joy and gratitude, I humbly request our principal, Dr. Sister Jyoti, Nirmala College, Ranchi, to address the closing ceremony. Over to our principal.
Thank you, Nikki. Am I audible, Nikki?
Yes, ma'am.
Yes, sister, you're audible.
A very good afternoon to you all. Respected Vice Principal, Sister Shobha, our esteemed resource person of the final session. In fact, it was the first session and it became final session. Dr. J.P. Pandey, Senior Scientist, CTRTI, Ranchi. Respected HODs, Dr. M.R. Rani Seraphim, Dr. Anubhuti Singh, and entire team of Botany and Zoology Department, all faculty members, and my dear students. Someone has rightly said the difference between a subliming block and a stepping stone is how high you raise your foot. I certainly hope and believe that today's workshop must have surely raised your level of understanding and learning. Let me tell you, success is never instant. It is always gradual process because it gets nurtured by your trials to attain the best in a proper sequence. So never quit learning, and you will see that you are consistent on the path of progress. Permit me to tell you that we all have worked very hard to make our institution as one of the premier center of learning, a college with potential for excellence, accredited by NAC with grade A, and still aiming higher level. Hence, we have set high standards and expectations of our students as well to achieve and behave their very best. Our dedicated staff and supportive facilities enable us to deliver our programs and activities in an environment that is intimate, friendly, and attuned to every specific educational need of our students in today's perspective. Today, I feel overwhelmed and proud to witness this online workshop, very specially dedicated to the students on the topic exploring the science behind sericulture, endocrinology, and ethnobotany. I deeply appreciate the initiative taken by the Department of Botany and Zoology, Nirmala College, Ranchi, to keep the students in tune with their subject. Moreover, to have passion for learning their subject. No doubt that this will help them to cultivate likeness towards their subject. My wholehearted congratulations and best wishes to the organizers, very especially taking the lead, that is Dr. Emma Rani Seraphim, Dr. Anubhuti, and the entire team of Botany and Zoology. I hereby congratulate each and every one of you for your efforts and participation in making it a successful learning experience, my dear students. I wish to see the same vibrancy in the days to come in every facet of life, and wish that all be blessed with success. I wish all your future endeavors as well. Stay blessed and stay safe. May God bless you all. Thank you.
Thank you, sister. Thank you very much.
Thank you, sister. Now, may I request Dr. Anubhuti Singh, Department of Botany, for concluding this workshop.
Thank you, Nikki. Respected Principal Dr. Sister Jyoti, Vice Principal Sister Shobha, Honorable Resource Persons Dr. J.P. Pandey, Dr. N. Venkatappa Rao, Dr. Purvi Saikia, Dr. Emma Seraphim, HOD Department of Zoology, my colleagues and students. Good afternoon to you all. We have come to the end of all the 3 sessions of our workshop. Definitely, these 3 sessions were highly informative and helpful in giving you insight to some of the very important topics that is a part of your CBCS curriculum. The motive behind organizing this workshop was not just to impart education on certain relevant areas, but also to develop scientific aptitudes among students to understand and learn the methodology of exploring science that is a basic part of our life. I believe our efforts would have surely helped you all in fruitful learning. I also wish to express my gratitude to our principal for providing constant support and encouragement. Thank you, sister, and thank you all.
Thank you, ma'am. It's my privilege to extend a very hearty vote of thanks for sharing with us your findings today. All three sessions were a very helpful one. Experience, and it is going to help us in our academics as well. Gratitude to our principal, Dr. Sister Jyoti, Nirmala College, Ranchi, for her guidance and tiring effort to make this workshop. I thank our Vice Principal, Sister Sobha, Prakash Pandey, for this wonderful session of Sericulture, which is enhancement, and it is going to help us in our. I thank Dr. N. Venkat Apparao for his ceaseless effort in endocrinology. I thank Dr. Purvi Saikia for her tireless effort to make us ethnobotany. My sincere thanks to Emma R. Seraphim, HOD, Department of Zoology, for taking all the pains for arranging this workshop. I also extend my gratitude to Dr. Anubhuti Singh, Department of Botany, who has motivated us and who has taken the effort to make the department participate.
इसके बाद anthem.
Each and every one of us are grateful to our HODs. A heart of thanks to our mentors, our teachers, Dr. Indu Kumari, Dr. Anjali Smita, Dr. Veena Pani, and Dr. Manisha. And most importantly, I thank Mr. Aman Kumar Jha for tech. Also, I would like to thank Rutvi Kumari, Ashwarya Bara, Neha Kumari, and Khushi Kumari for possible ways. Lastly, I would like to thank my dear friends, without whose participation this event could not be a success. Now, I request and all my dear friends to arise for national anthem.
जनगणमन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे जनगण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
To an end, thank you each and everyone.
End कर देना ना meeting.
Thank you, each and everyone. Thank you, dignitaries and students, for being with us. Thank you very much.
इसको हमको कैसे दे पाइए? एक्सेल शीट में दीजिएगा।
हाँ एक्सेल शीट में दे देना।
इसमें दे दीजिए।
31 आया है अभी तक।
यही रहा होगा ना?
हाँ अभी तो यही रहा। 31 रिस्पॉन्स है।
अभी 31 ही आया है। तो बंद हो गया, फिर मान लो लिंक को नहीं मिलेगा तो कैसे करेगा?
यूट्यूब पे तो है ना।
हाँ
हाँ, लेकिन अगर कोई सोचा हुआ कि हम यहाँ पर से ही करेंगे जूम से, तब उन लोग फंस जाएगा, तब वो लोग नहीं कर पाएगा।
जो है आने दीजिए थोड़ा देर। एक सर्टिफिकेट का तो इसमें चक्कर नहीं है, अभी अटेंडेंस के लिए। यूट्यूब पे कैसे पता चलेगा?
अरे यूट्यूब को भी ये करना है।
अच्छा, इसको बंद करना है ना।